Ano'i me ke aloha kākou, aloha kakayaka. Mahalo everybody for joining us this morning. My name is Mina Viditua Jr. I am the newest program coordinator with the Pacific Internship Program for Exploring Sciences, and I'll let my hoa hana introduce himself.
All right, aloha kakahiaka. Thank you for joining us today for our 2021 PIPES Symposium. My name is Devin Aguiar. Along with Mina, I'm one of the program coordinators for the PIPES internship program this summer. We had a blast, so a little backstory. Both Mina and I are both former interns, so now we're seeing the other side of the internship program, and I gotta say, I give props to the coordinators before me because there's a lot of work and a lot of dedication that goes into it, but we had a blast this summer. We learned a lot. We really got to connect with our cohort, our haumana this year. It's been a blast this year, and thank you again for joining us. We're really excited to bring you the presentations we had today about the projects that these interns worked on, and yeah, mahalo for joining us today. I'm going to hand the mic off to Sharon Ziegler Chong, our director, who will be giving a little opening speech for today.
Aloha mai kako. It is weird. We have all of our audience on camera, and we have all of our interns here in person. So we're really glad that we are able to be here together, and mahalo for joining us online. PIPES has been going for many years. This is their 28th program year, and that is pretty inspirational and pretty amazing when I look at all of the people who are in this room and online and across our islands that are really engaging here. So we have a program all for you this today. People are excited about giving those. PIPES has evolved. We are focused. I'm gonna read it because we've spent a lot of time in looking at our developing our purpose and mission, because it has evolved. So I just wanted to read it. So our purpose is to empower students to identify their role and pathways to aloha aina. And the mission, the way we do this, is to cultivate experiences and relationships that foster self-exploration and mentored learning by immersing students in the values and practices of Malama Aina. And these interns this year, you'll see in these presentations, that's what they've done. And we've been really grateful to work with them. And Devin and Mina, I have to hand it to them. Wow, I think we pulled it off. In the time of a pandemic and where these guys' worlds are so weird, think about that for those of you who are established professionals trying to start your career out in this world. So I really mahalo all of the interns for all their experiences and energy, and especially all of the mentors and host organizations that helped us provide these experiences, as well as obviously the University of Hawaii at Hilo, where this program has been running this whole time, and they've supported us with all so much here, and our sponsors, Ha'oli Mauloa Foundation and Kamehameha Schools. We are excited, and I should also mention we have the National Science Foundation, which has been a very important sponsor of our efforts. So we're gonna start this off. I am going to hand it to Mina, and then right before we start, I'm gonna add a few things too for the interns and for the audience as well. Mina, do you wanna start us off?
Mahalo, Sharon. So before we start, I actually want to do a small pule for us to bless everybody in the audience and everybody who is also here joining us in person. So, e ho'omalu kako, pule kako I ka pule e na o makua, I na mapopo o ko ka hualela e komo mai. E na o makua mai ka lahiki a ka lakau, mai ka ho'oku I a ka hala wai, na o makua I a ka hina kua I a ka hina alo. Ia ka akau I kalani, o ki ha I kalani, o e I kalani, nululu I kalani, ka aholo I kalani. E ia ka pula pula o ko, o ka ohana o Hawai'i. E malama o ko ia mako, e ulu I kalani, e ulu I ka honua, e ulu I ka pai aina o Hawai'i. E ho mai ka ike, e ho mai ka ikaika, e ho mai ki aka mai, e ho mai ka mapo pono, e ho mai ka ike papalua, e ho mai ka mana. A mama, ua noa.
Mahalo, Mina. Okay. So we are going to start on the hour at 9 o'clock. I don't know what we thought we were going to talk about for 15 minutes here, guys. We always have some ideas of things, but then they always kind of whoosh, go out the window. But just a few things of protocol. So for the interns, they don't know this is coming, but for the interns, because we only have you all in the audience in person, it is your job to come up with one question for every speaker. And we'll be able to entertain probably one, maybe two questions, but you guys need to generate those questions. For people online, we will be monitoring, and so me and Deb, we just need to make sure that we're monitoring for questions online too, and we'll be asking those as much as we can. For the interns, you're going to be standing up here, the camera will be on you, and whenever there is a question asked, you guys need to repeat the question so that the audience, the online audience, can hear what that question is. So that's very important. And you'll hear me going, if I go, that means that you haven't repeated the question. So it will be, you'll have your presentations. People have worked really hard on these presentations, putting them together. And then we will have a question and answer. And if there are questions that are remaining, we'll get back to you. We'll share those questions that are posted online with the interns too, and then they can get back to you as well. Are there any questions? I can repeat the question. Any question? No, there are no questions about today. Yeah, wonderful. All right, I think we're going to pause just for a little bit. And I also want to mahalo Kahaku'ula for giving us this beautiful space. I'm sorry that the audience here online cannot share this space with us. This is such an important space for us to gather, and we're able to gather here. And again, my apologies that you're not able to join us here, but thank you, mahalo nui for everybody's work here in providing us this space to gather. So I think what we will do is give 5 minutes for everybody to just shake out the jiggles and the nervous things, and we will be a start promptly at 9. Yeah, okay, mahalo.
All right, aloha mai kako. Without further ado, well, we have our first speaker today, Alyssa Matthews, and she'll be giving us a really good talk about her experience this summer. Take it away, Alyssa.
Okay, aloha everybody. My name is Alyssa Matthews. My internship this summer was with the Spatial Data Analysis and Visualization Lab at UH Hilo, and my mentors were Ryan Peroy and Tim O'Sullivan. So my project was using high-resolution imagery to assess rapid ohia death tree mortality associated with ungulate presence or absence. So much is still unknown about the spread of rot, but a possible explanation is ungulate activity. So ungulate activity such as trampling, stripping of tree bark, and wounding trees are all potential ways that rot is spread from tree to tree. However, a solution to this is to implement fence lines in order to keep ungulates out. That brings me to the research question for my project, which was if ungulates were fenced out or exclosed from a forest, would that fenced forest have less rot-infected ohia trees than unfenced forests? So to answer this question, I looked at satellite imagery in order to cover a wide area over several sites on Hawai'i Island. The imagery was cataloged based off of the recency of the imagery. The image had to have little to no cloud coverage, a high spatial resolution, and it had to cover my sites of interest, as well as the imagery had to be multispectral with near infrared and red bands in order to be able to perform NDVI calculations. So after narrowing down what images I wanted to use, I went into image processing and pan sharpen the imagery to get a better resolution. And just to give you an idea of what the imagery looks like before and after pan sharpening, here's the original satellite image, and here it is after being pan sharpened. And here's just a zoom in side by side, and as you can see on the pan sharpened image in the right, you begin to see some individual trees, which gave me more to work with. So my sites of interest were chosen based off of the imagery that I had available, and the sites of interest were known to have fence units and were known to have rot-infected trees from previous helicopter surveys done in those areas. So I ended up with 8 sites across Hawai'i Island with 19 satellite images that covered areas over several different dates so that I could look at changes over time. And here's just an example of what one of my sites might look like. The yellow outline represents a fence unit that hopefully is ungulate-free, and the red dots represent suspect rod-infected ohia trees, which we know from the helicopter surveys. So after choosing my sites and pan-sharpening the imagery, I began the process of using NDVI, a vegetation index, to gather information on the overall forest health in my sites of interest. So NDVI is an index with values ranging from -1 to 1, with higher values closer to 1 indicating healthier vegetation. It's basically used as a proxy for overall forest health. So the NDVI image comes out as grayscale with the darker pixels representing less healthy vegetation. And you can sort of see that here in the bottom right corner of this image. So on the right, on the bottom right corner of the image in the true color, you can see that that's either probably exposed soil or possibly a cluster of unhealthy trees. And in the grayscale NDVI image, that comes out as darker than the surrounding healthier vegetation. So after doing that, I created these ROIs in and outside of fence lines to be able to compare the mean NDVI of the unfenced and fenced forests. So after that, I ran stats on them to find the mean NDVI. And this is what came out of that process. So an interesting thing happened where all of my mean NDVIs for the interior and exterior ROIs were coming out as statistically different. However, for values that were really close together, it was still coming out as statistically different, even if they had very little differences. And this was likely happening because if you look at the two far columns on the right, my population sizes were so large, leading to little differences coming out as significant. So in order to address that, I had to look at effect size. So effect size is a number that measures the strength of the variable in differentiating two populations' characteristics. Basically, I wanted to see if these statistically significant differences were meaningful or not. So we found the effect size for each site using Cohen's d equation. Basically, the effect size would tell us if the fence lines were effective in maintaining healthier forests or not. So as you can see here, effect size is a range. So a small effect is around 0.2, medium effect around 0.5, and a large effect is around 0.8. And so we calculated Cohen's d for all of our sites. And as you can see here, most of our sites had a positive effect, generally meaning that fenced forests were healthier than unfenced forests. However, we were seeing that there were some gaps in the effectiveness of fence lines over time. For example, North Ala'a, it had a close the effectiveness was close together over time, but for South Ala'a unit, there's this gap in effectiveness at different points in time. So this is just another way to illustrate that the numbers on the left come from satellite imagery taken earlier dates on time, and the right are later dates. And so for most of the sites, there aren't too many huge differences in effect size. However, for the South Ala'a unit outlined in red, it went from a large effect to a small effect from 2018 to 2021. And we do have history on that site to explain the difference here. So we actually know that this particular unit, the Koa unit, had been breached by ungulates in 2020. Prior to that, there weren't very many suspect rod trees within the Koa unit exclosure, but after the breach in 2020, we began to see a larger amount of suspect rod ohia trees over time. And this could be a possible explanation for why some of these areas experiences those gaps in the effectiveness of fence lines over time. So as we know from the Koa unit, we can address that the fenced areas don't always mean that they are ungulate-free. So the more information that we have in these areas, the more we can see why the differences in effectiveness of the fenced lines occur over time. Furthermore, while NDVI is representative of overall forest health, it's not exactly a measurement of rod itself. So to address that, another method I began to explore is using a supervised classification on ENVI, a maximum likelihood classification. So I used a maximum likelihood classification to measure red pixels that were representative of red crown ohia trees. And maximum likelihood classification classifies pixels based on the highest probability of that pixel belonging to a certain class as determined by its mean covariance. So I ended up with these different land cover types and different classes based on the land cover types I had available in my imagery. So just zooming in and giving you an example of what the classification looks like. Here's the original RGB image, and here it is zoomed in. You can see that those are individual red crown ohia trees. And here's the classified image I ended up with. So in this image, although it did pretty well, it looks like there is some confusion between the different land cover types. A prominent one I kept on seeing was a confusion between bare soil and the red crown ohia trees. And so I collected data on that, the red pixel percentage, in a similar process to the NDVI data collection, in which I did those ROIs in and outside of fence lines to compare the pixel percentage in and out of fence lines. So as you can see here, it does appear that the interior red crown percentages were lower than the exterior red crown percentages. However, there's still the question of whether or not these differences are meaningful or not. For the large difference, like 3.6 for the South Alakoa unit, we can probably assume that those differences are in fact meaningful. So after I did my classification maps, I had to perform accuracy assessments on them using validation ROIs and a confusion matrix, basically to see how well my classification maps did. And as you can see here, it appears that 100% of the pixels were correctly classified as red crown trees, but as we saw in my classification map image, that was not the case. So probably adding more validation points would help me better understand my accuracy assessments. And a common misclassification I was having was a misclassification with bare soil. As you can see in the bare soil column, it was misclassified with a lot of different other land cover types, but in particular, most prominently, it was the red crown class that was getting misclassified. And a possible reason for that is, as you can see here, here is the spectral profile for a red crown ohia tree, and here's a spectral profile of what looks to be bare soil. So the similar spectral profiles was a common confusion in the classification efforts. So to wrap up here, I found that overall forest health was generally higher in the fenced areas, and again, with further classification efforts, possibly collecting more training pixels and validation pixels would help to create more accurate maps that would get a better read of the presence of rod in and out of the fence units. However, for further research, more information about the management of these fence lines and the presence of ungulates around them would be necessary to understand the differences in the fenced and unfenced forests. And with that, I would like to thank my mentors, Ryan Peroy and Tim O'Sullivan, for their help and guidance, Brian Tucker for the satellite imagery, NSF, the State of Hawaii, and the National Park Service for funding the project, and mahalo to everybody for your time. Oh, sorry, any questions? Yes.
What program did you use to make the identification system, and was it really difficult for you?
I used Envy to create the question was, what program did I use to make the classification maps, is that right? And how difficult was it? So I used Envy to create the classification maps, and it wasn't so much difficult as it is time consuming, because I'm picking out these individual pixels that are representative of the actual land cover types, and it takes a while for it to come out well, and it takes a lot of pixels for it to come out well. Thank you. Yes. What was the total area of our study? We don't have, I don't think I have an exact number for you, but yeah, we did have the 8 sites across the west and east side of the island. Yes.
How did you choose your sites of the study?
How did I choose my sites of the study? So basically, a big thing was can I actually see my site? Because of things like cloud coverage and the quality of the picture and things like shadows and all of that. Those definitely played a role into what I could work with. And again, I had to know, or we did know that those areas had fenced units and that there was rod present in those areas. Yes. Did I personally get to see the sites? I did not get to see all of the sites, no. Yeah, Sharon?
So where does this take you now? Is this making you more interested in this kind of technology or more into the conservation question?
So the question was, where does this take me now? Am I more interested in the science behind it, or the technology behind it, or the conservation? I think probably both. Doing this was really interesting. It was interesting to see what kind of issues kept on popping up with, again, like what imagery could I use? Was it appropriate to use it? That kind of thing, yeah. Any other questions? Thank you.
Aloha mai kākou. Aloha, my name is Lenny Helekahi, born and raised in Papakolea. For this internship for PIPES 2021, I had the opportunity to work with Io, the Hawaiian hawk. I'll give you some context to my research. Some things that we'll be going over is the intro to Io, some cultural significances, background data and studies that I use to develop my own study, my methodology, data collection, of course, results, discussion, and of course, our conclusion. Some introduction to eel, some scientific background. So eel, also commonly known as the Hawaiian hawk, has an estimated lifespan of 17 years. It lives in elevations of about 30 meters above sea level to 1,700 meters above sea level, which is 100 feet to 5,577 feet. The habitat that it lives in is mixed. They live in non-native forests, native forests, open canopies, closed canopies, agriculture and pasture lands, but most importantly to remember is they like to nest in ohia trees. So anywhere where there's an ohia forest or ohia trees is possible eel territory. Their diet used to consist of native birds, but from what we've been discovering, they've actually changed their diet to feast on invasive species of birds. For example, minor birds, northern cardinals, and Japanese white-eyes, which are commonly found throughout Hawai'i. Oh, and don't forget the rats and mongoose as well. For mating, they are monogamous. Had a hard time seeing that for practice, which basically means they have one mate at a time. They usually have one egg per relationship, but it is also known that if they get into territorial conflicts with another eel, they will abandon their nest to find somewhere else to start over. Some cultural background. So if you've seen my HEC presentation, Io is commonly known or associated with E, this unknown supreme deity of Polynesia. Upon further research, I discovered that David Malo translates Io as Hawaiian as hawk owl, which is funny because in science, a hawk and an owl is two different animals, but in the Hawaiian perspective, they were seen as one and the same. Pueo was in fact Io's counterpart. We see this a lot in Hawaiian culture as opposites, for example, Kana and Kanaloa, Lono and Ku, and also birthing pairs coming from the Kumulipo. When we often think of Pueo, we think of Auma Kua or family guardians, and we also think of their role in Mo'olelo as having these supernatural powers. For example, bringing people back from the dead in the tale of Kahalaopuna or the Rainbow of Manoa. Most commonly, you hear them as messengers that warn of danger. For example, a lot of them will stop in front of vehicles today, and they will refuse to move until some time has passed. Usually, those who are caught up in those events say that, oh, there was an accident further down the road, and the owl helped me not get involved with the accident. Pueo is also, upon research, is an extension of E. E being the master and Pueo being the messenger. The example that I gave to my mentors was kind of like God and angels. God being the one to have his heavenly will be done, and him sending his angels as the ones to fulfill his needs or his duties. The reason I think we don't hear about E anymore is because we credit that supernatural ability to the Pueo instead of E. So that is why we no longer hear of E or Eo. Some background data. So I use two primary resources in my development of my driving question. The first is taken from Gorson. P. Marcos Gorson has this study entitled Abundance, Distribution and Population Trend of the Hawaiian Hawk, and it is dated from 1998 to 2007. This study did not necessarily take place within those years. What happened is a study from 98, he basically revamped it in 2007. The purpose of that study was to survey the population of hawks on Hawaii Island. So the method that he used was playbacks of adult eel, spaced 1 mile apart, and then observations were recorded. For his results, a total of 545 sites, he's seen 185 eel. So approximately 1 eel for every 2.95 sites. The second primary resource I used was from Burnett. Jessica Burnett conducted this study on the continent, and the title of her study is Songbird Distress Calls as an Improved Method for Detecting Red-Shouldered Hawks. This took place in January of 2016. The purpose of her study was to use a variety of playbacks to see which call is most attractive to red-shouldered hawks. Her results, raptors, not just hawks, but birds of prey, were observed at 23 out of the 124 sites, with most seen during the songbird distress call. The reason this study is important to me, though it does not occur in Hawaii, is because the red-shouldered hawk is the most closely related hawk to our eel or Hawaiian hawk, which is why we used it. Methodology. So the question that I wanted to answer was, what calls do eel respond to most frequently? The way we did this was we surveyed 24 sites across the Hilo and Puna districts, and we conducted our data collection between the hours of 9 to 5. We did this because that is when eel are most active during the day. So we used a bunch of different calls. We used an eel call, a prey bird call, which came from a northern cardinal, and a control, which is no playbacks, simply kilo or observing. To basically determine our sites, we used ebird.com. I'm sorry, that should be ebird.org. eBird is a site where the public can actively track your GPS location and tell the public where they have seen eel or where they have spotted eel. Some of the criteria for the sites were they were at least 1 mile apart per sighting, not in a state park or on private property to avoid permits, and they were also along roadsides for convenience. How our methodology was developed was, as I mentioned before, we took a combination of Gorison and Burnett's studies. So from Gorison, we took his eel playbacks, his 10-minute time intervals, which means he played a call for every 1, 4, and 8 minutes out of a 10-minute interval, and his 1 mile apart sites. From Burnett, we took the idea of using a prey bird call for one of our methods. The purpose of my research is to further the research of eel in Hawaii because, again, not much is known of eel. And for future research, I mentioned previously at HCC, there will be GPS tagging to see habitat use and range of eel. Data collection, super simple. Upon arriving at a site, we disembark from our vehicle and set up our equipment. Our equipment included Bluetooth speakers, binoculars, Kestrel rangefinder, GPS, and our cell phones. We'll take initial observational data, for example, weather, temperature, wind speed, and a GPS point. Then we would silently observe for approximately 1 minute to see the baseline of the environment. So for example, birds that are already singing or birds that are not singing according to our call. Then we'd start our timer and do our playback. We'd stand in the vicinity of our Bluetooth speaker facing different directions in order for us to observe. And then we'd start our timer. If an eel was encountered, the timer would stop and we would record our data. If no eel was encountered, the timer would complete its 10 minutes, record our results, then we'd be off to our next site. Something really cool that I've learned from my mentors is how to use Survey123. Survey123 is an online platform that is used to create surveys. It's a super complicated tool that is used by science, but once you basically do the coding and whatnot for your survey, it can be used on your cell phone without the use of like internet. So you can bring up, it looks like a Google form, you fill out the form, and then when you don't have internet in the field, it saves it. And then when you come back and you have internet, you can upload it to, the internet. The purpose of this is it's super easy to use once you get past the coding part. You don't need paper in the field, and you can do a lot of different things representing the data. So here's my results. Out of 24 sites, we've only seen 2 eel. And the two EO we encountered were both doing our control method, which means no call. Here's a map of where we surveyed all of our sites. So our first site is located on the very top in Hakalau near Akaka Falls, and our second is located in the residential area of Hawai'i Paradise Park. Both sightings were again under our control method of no call. We simply waited and we seen EO there. My discussion, developing a research project. This is not based off of the experiment itself. This is based off of the experiment as a whole, or the experience as a whole for PIPES. Developing a research project was kind of challenging because my mentors gave me free rein of what to do or what not to do. And like I mentioned, I need some sort of direction in order to work towards something. Originally, we were going to let Mo'olelo inform us of what we needed to answer for EO. But as I've mentioned, we kind of hit a dead end when EO fell off of worship by Polynesia. Time constraints. Because PIPES is a 10-week program, me and my mentors were trying to come together to see what kind of project we could complete within 10 weeks. We didn't want to do anything too large where it took too long and we wouldn't have results, but do something substantial that would impact the community. Because of the time constraints, a lot of different factors played. For example, accessibility. We limited our sites to roadsides in order for us to survey as many sites as we could without having to get out, travel miles into the forest, travel miles into state parks on private property, because that all took time. Permitting was another factor. Doing the research with my mentors, I've discovered that permitting is quite a process when you're trying to do research, and permitting can hold up research for long periods of time. So again, in order to not deal with permitting, we did not do anything illegal. Presenting a completed project. According to our PIPES coordinators, it was okay if we came to our symposium and did not have something completed or did not have substantial data to share. But I talked it over with my mentors, and I think personally, I felt more comfortable presenting something completed. I mean, my data speaks for itself, but it is a completed project, and I feel confident that I completed it. EO habitat. Scientifically, we know that EO habitat spans for more than a mile. In fact, it's several miles. It could be 20, up to 50. But of course, because of time constraints, and we were working with Gorison's study of 1 mile apart, if we didn't follow that, we probably would not have completed our project. Cultural wanderings. In the very early stages of my research, we had these very culturally enriched questions that would not be answered by science, or that could be answered by science. My mentors were a huge fan of what we found culturally in terms of Hawaiian culture research. So one of the cultural wanderings that we had that we did not get to test was what would happen if we played a pueo call in the presence of an io. As I mentioned before, they're opposites of each other, their duality. Would they recognize each other in that same way? It was something that we ultimately had to abandon because of time constraints, but if anyone would like to test that for me or share that with the community, by all means. In conclusion, io exist on Hawai'i Island. They do. We've seen 2 of them. It is uncertain if prey bird calls are effective at attracting io, but again, a larger study is very necessary. For future research, again, stay tuned to the up-and-coming io studies that will be happening very soon in terms of GPS tagging and habitat research. Personal conclusions. This was an idea that my mentors put in for me. So some likes and dislikes because that is what the purpose of this internship is supposed to be. I enjoyed the field work and I enjoyed making connections with my mentors and learning a whole bunch of different skills that I never learned before. Dislikes, the politics of it. Conservation takes a lot of you, it takes a lot from you, and it doesn't give much back. But a lot of different cool sayings were said on this internship, and one of my favorites was, for conservation, we do it for love, not the likes. For my ka'aua personal journey, which has been a huge theme for PIPES, Again, I've never imagined this for myself. As a Hawaiian Studies major and not being good at math or science, options are very limited. But because of our wonderful coordinators, our mentors, they showed me that there are more to life than just teaching Hawaiian Studies, Hawaiian language, but you could have a career in conservation. In conclusion, again, I would like to mahalo our PIPES coordinators, our program, Bryce Masuda and Amy Durham, my mentors, and all the other people who contributed to my research. And like all conservation, aole po. Are there any questions? Yes, Uye. Because E fell out of favor, will I put it into my personal life or make it known for future, or for the future in general? That's a very difficult question. In the Hawaiian perspective, things, for better or worse, die for a reason, or things are forgotten for a reason. And the resurrection of certain things is not always good. In terms of deep Hawaiian culture, I think I'd have to research a bit more or ask people who are more experienced than myself. But in terms of io as relating to io, the Hawaiian hawk, I guarantee that information should be put forward and should be put forward into the future of conservation. Thank you.
Mahalo, Leonard. Awesome presentation. Next we have Lehua Patnal. Mahalo.
Aloha mai kako. My name is Lehua Patnod, and I'm a student here at UH Hilo. I'm originally from Kahalu, Oahu. Oh, kalamai. I'm originally from Kahalu, Oahu, but I've lived here long enough to call Hilo my home. This is my first internship with the PIPES program, and I was fortunate to spend my time with an amazing organization, Hui Malama Ike Ala Ulili, or Hui Mau, located in Hamakua. Their work focuses on reestablishing systems that will sustain their community and reduce its dependency on external resources. Ho'u'ulu'ai is one of the goals that Hui Mau focuses on and where my presentation begins. I would like to acknowledge these organizations for allowing me the opportunity to be a part of this wonderful and educational experience. Mahalo to the University of Hawaii at Hilo, Hau'oli Mauloa Foundation, Kipuka Native Hawaiian Student Center, Kamehameha Schools, Pacific Internship Programs for Exploring Science, National Science Foundation, Louis Stokes Alliances for minority participation, and last but not least, the crew of Hui Malama Ike Ala Ulili, and a special shout out to my mentor, Cody Sales Kalani, who is a former PIPES intern as well. And I would also like to acknowledge Kahaku'ula o Ke'elikolani for allowing us this space to share our Ike with each other and with each other. Mahalo and aloha. My hana was located in the moku of Hamakua, Kihiloa, Hamakua with a long corner, or known simply as Hamakua by most. Hamakua Kihiloa, as its name implies, refers to its large land base that touches each moku of Hawaii, except for Puna. Its boundaries begin at Ka'ula Gulch near O'okala on the Hilo side and stretches across the rough and irregular terrain to its Kohala boundary at Honokea, and then extending upwards, Mauka, over Mauna Kea, to the summit of Mauna Loa at Pohaku o Hanalei. A little brief history of Hamakua. The Aina in Hamakua has been through a lot of land management changes. The landscape in Figure 1 would represent a healthy forest environment. You can see the kukui growing along, and I think in the corner, to me, it looks like that's some banana, some mai'a growing as well in the left-hand corner. Before the era of sugarcane, this aina was covered by kukui. There were 5 groves that were famous, and Hamakua was one of these areas. Hamakua was also known for its planting technique of pa kukui, and it was implemented in the forest groves here in Hamakua. Pa kukui its literal translation means candlenut enclosure. This method was implemented in a concentrated area of kukui where they were felled and then allowed to decompose, thereby creating rich organic matter for the planting of kalo or taro. Large holes were dug about 10 feet in circumference and then about 3 feet deep, depending on the laborer, and then filled with the kukui leaves and then covered with soil and left to decompose. They would also leave branches sticking out, so the stem would stick out, and when they would come and check to see if it was ready to plant, they would pull the branch out, and if the leaves were decomposed, it was ready to kanu. 1840s sugar plantation era, and it had a strong foothold in Hawaii's agriculture, as well as the socio-political economy side of it as well. But as the price of sugarcane, of coal, was cheaper to export in other areas, there was a decline, but it lasted for 100 years plus. Figure 1 here, we show the eucalyptus groves, and that came in after the sugar plantations. In 1978, there was a joint study between the USDA Forest Service's Pacific Southwest Research Station and the Sea Brewer Company Limited, and they established a joint research for managing short rotation, 5 to 8 years, to determine if the eucalyptus was a suitable source of bioenergy. Test plots were located along the coast of Hamakua, as well as on the dry side of Ka'u. In Figure 2 is a picture that I took a couple weeks ago of a gulch in Hamakua. And as you can see, there is eucalyptus, kukui, we have mai'a growing, and then the guinea grass in the front. But just showing what the natural landscape used to be, because these gulches were so steep and it was hard to cultivate there, it was left untouched. So there are a lot of native plants still growing in these gulches today. Okay. And that brings me to my host organization, Hui Malama Ike Ala Ulili, or Hui Mau. Established in 2009, Hui Mau's founders are comprised of ohana from the community. They are dedicated to create kipuka, safe and regenerative spaces, by reincorporating place-based ancestral knowledge, also connecting to Aina through stewardship, and reestablishing and strengthening pilina for all generations within the community in order to grow and thrive in Hamakua. Their hana began in the ahupua'a of Koholalele, so in Figure 1, it's highlighted there in red. And they began to establish restoring aina and abundance in the Makai region of this area. My work was focused at Kahua Ho'ama, and it is a 5-acre aina restoration and farm site located in the Ili Aina of Waipunalau, in the Ahupua'a of Pa'awilo, in the Moku of Hamakua. The primary purpose of this wahi is to create a safe community space for youth and ohana to gather and learn together through malama aina and to reestablish food sovereignty in this ahupua'a. There are 4 pas in this kahua ho'ama, the first being pamua. So in Figure 1, we're showing the eucalyptus grove, as you can see. So this is what it was prior. The red roof on the top corner, right corner, is Hale Pa'uilo, where Hui Malama Ike Ala Ulili is located. And so right up above there is that eucalyptus that they were able to open up and restore. Figure 2 and figure 3 is what it looks like now of this area. We have Pahanai, where there are animals being raised. We have Napua Wahine here. We have Kalamansi and Jabong, our goats. There were also sheep and then chickens as well. We have Pahale in figure 1, which is actually in the background, and that will eventually be used to establish or to erect a hale for community to come, to share, to gather, to eat. Pakukui is the area that I focused my work on, which is figures 2 through 4. And as we can see, they've established kukui, ulu, kalo, mai'a, to start restoring this aina back to what it used to be. Part of my project was pakukui, so we did a little replication. Here's our kumukukui that we used, mahalo. Figure 2 is the area that we scouted. We're looking at, for time constraints, we went ahead and we broke up the kukui, and we put that into a little puka, not as deep as 3 feet, but good enough to serve this purpose. We went ahead and covered that with soil, and then just so it would decompose quicker, we put a weed mat over that. We did get some preliminary soil analysis. So on June 23rd, we were able to turn in a soil sample. Starting pH was 4.9, and then we're looking at the calcium, magnesium, potassium, and phosphorus, parts per million. We did send in a second sample, which reflects figures 1 through 3, the after digging it up with the kukui. So you can see how within that small period of time, from June 23rd to July 20th, it decomposed rather quickly. So that was good for us. And so we're just waiting for those results to get in and then be able to kanu some kalo. So also part of my project was growing food, was able to implement some beds of uala, kalo. Figure 2 is the beginning of the kalo patch, and then in figure 3, it's hard to see because of the contrast, but we have the kalo beginning to grow. Other plants on the aina, we have ipu, mai'a. Figure 4 shows ulu and kukui growing together, our harvest, and then figure 6, just sharing that with their community with one of their kitchen work days, community days. Mahalo. Ninau? Aye. Definitely, definitely. And I encourage all of you to, oh, excuse me, will I continue to do work with Hui Mau after this internship is pau? And I definitely plan to. I am now aware of this area and have put in time and would like to continue that as well. And I encourage you all to go ahead and check out their website and even see when they have Ohana days, you know, to volunteer or even donate. Yes, Drew. Did the kukui leaves end up decomposing? Yes, but not fully all the way, but most of them did, yes. It was about a month where we did that, because we put that in June 23rd, and then we did another soil sample about July 20th. Thank you. Amy? Definitely, you know, I will try to use that depending on the area that I'm in. Oh, the question was, will I implement pakukui in my area? And I will definitely try to replicate that to see if it would be feasible in the area that I live in. So I'm in Kaumana, where it's really wet, and I have one kukui tree growing. So I need to plant more so I can fell them and do this replication. Mahalo. Mahalo.
Aloha mai kākou. Okay, this is weird to hear my voice and hear the mic, so we'll get used to this together. Mahalo mahalo to everyone for being here today. My name is Darian, I'm from Kalihi, Oahu, and I will be sharing my PIPES project, which was Wailepo assessing connectivity of on-site sewage disposal systems to Hilo's shoreline waters. So as with many words in Olelo, Hawaii, there are multiple meanings for Wailepo. The meaning that I'm using for this project is dirty or excrement water, which is sewage, and that's what we're going to be talking about today because that was my summer. So let's start with sewage pollution. Globally, sewage pollution has affected coastlines as it releases high concentrations of inorganic nutrients such as ammonium, nitrite, nitrate, and phosphate, as well as endocrine disruptors, suspended solids, heavy metals, pathogens, and other toxins. So this is all big words for bad stuff. Bad stuff in the sewage going into our waters. Over 80% of the world's sewage is released into the groundwater to the shorelines without treatment. That number goes up to 95% in some of the least developed countries. So with all of this bad stuff in the water, this can have an impact to coral health as sewage pollution is getting into the water, causing a nutrient increase and leading to macroalgal dominant reefs, which again means that there's a lot of algae growing and it's slowly drowning out our corals. This can also have an impact to human health. As with anything that happens in the environment, exposure to sewage in the water can cause skin and urinary tract infections, stomach virus, and other respiratory diseases. So in Hawaii, we have what are called on-site sewage disposal systems, or OSDS, which are essentially sewage management systems meant for your home, and this includes septic tanks and cesspools. So pictured here is a cesspool, which is a hole in the ground that collects all of the sewage from your home, coming from your drains and your toilet. All of this sewage comes through the rocks, goes into the water untreated, and slowly makes its way to the ocean with all of the contaminants that are in there as well. In Hawaii, wai, or fresh water, is such a valuable resource that saying it twice, saying wai, means wealth, or talks about wealth. So the fact that our lepo, or our excrement, is making its way to our oceans is a very big deal. And as of 2017, there was an estimated number of 49,000 cesspools in the entirety of Hawaii, and 53 million gallons per day of sewage discharge making its way into our groundwater. On Hawaii Island, we have 8,700 cesspools in Hilo, in Hilo alone. Hawaii Island has the highest percent of risk to coastal waters for OSDS impact. And in 2017, the legislature passed Act 125, which requires the replacement of all cesspools by the year 2050. Hilo Bay is a priority 3 area, meaning that there is a potential impact on sensitive waters. So this area right up here. And Hilo, and more specifically the Keaukaha area, has very little to no water quality research available. Therefore, the purpose of this project was to conduct water quality data for the community to be able to use in future decision making of what to do with cesspools in their community. So the objectives for the specific research project that I assisted with was to determine the connectivity of cesspools and shoreline waters, as well as to eventually provide these results to the Keaukaha community. A personal objective, however, was for me to gain further technical skills and knowledge in the field and in the lab. So, in order to conduct this research, we conducted what are called dye tracer studies,
which is where you flush this dye in the little orangey container into the system and flush it down with water as into the cesspool here, and then it slowly will eventually make its way to the shoreline. While we're doing this over 10 hours in the day, we are also collecting samples at the shoreline. The dye that we use for this study is called fluorescein. It's a non-toxic organic dye, so it is okay for it to be in the water. For the following days, we collected water samples at the lowest low tide to ensure that we were collecting samples from the groundwater springs. We collected samples from 12 stations along the shoreline in the area that we conducted the study. So collecting samples at the lowest low tide means that we were out there rain or shine, day or night, even if it meant that we were out there at 2 in the morning. So that was a fun thing to do. And this was all just to be there once the dye finally did come out at the shoreline. Once the samples were collected, we measured for fluorescence in order to get data on dye concentrations, and we measured conductivity to get data on salinity. The most important thing with this part was that we had to conduct analysis in the dark in order to preserve the dye in our samples because it can be broken down by light. So this is one of my figures. On the x-axis, you have samples by date and time. On the y-axis is fluorescein concentrations. The colorful dots up here represent different stations. So orange will be station F, yellow station 8, green is station 9, and 7 with no dye is station 7. So we have the date of injection all the way to what would be your left. On the 14th, we saw that dye emerged on the 17th, which was 3 days later, and it peaked on the 19th at station F at 7.5 parts per billion. We also collected data on salinity. This was in order to see if our dye concentration data was affected by salinity or affected by the mixing of salt water as high tide comes in. As you can see, again, the x-axis is by date and time, the y-axis is salinity. The colors represent the same stations again. And as you can see, it stays mostly consistent for the entirety of the study. There are a couple of days at the beginning on the 14th and on the 19th where salinity is a little higher, and this is because we were collecting throughout the day, so at high tide and low tide, just to be sure that we were collecting the dye that we were seeing. So while also working on this research, I assisted with collecting data for other projects related to water quality in Keaukaha. In the field, I was able to talk with the community, learn how to collect water samples, and identify groundwater springs on the shoreline. In the lab, I worked on acid washing and bleaching equipment in order to prepare for sample collecting. I prepared samples for Clostridium and Enterococci and nutrient measurements, as well as filtered water. So, from the figures that we looked at, we saw that dye emerged at 3 days on the shoreline and peaked at 5 days at Station F. This is consistent with another study that was done on this island in Puako, where they saw dye at the shoreline ranging from times of 9 hours to 3 days. We used the concentration data of the dye that we put into the system and the dye that we found at the shoreline to calculate the dilution. This is basically how much dye from when we put it in ended up at the shoreline. This dilution was 3.5 parts per million, which is equivalent to a couple drops of bubble bath in an average tub. And I don't know about any of you, but the idea that there's a couple drops of soap in my bathtub makes me a little uncomfortable with getting in the water. And something that's important to remember is this is the sewage from a single cesspool. There, again, are 8,700 cesspools in the Hilo area that is discharging sewage into our coastlines. So that's like 87,000 or 8,700 drops of soap in the bathtub. In the future, this research is still ongoing, but hopefully it will help to inform the community about what's going on their coastlines and help them get the required funding in order to decide what alternatives that they want to take switching from cesspools. Overall, this summer was a valuable experience for me because I got to work with these amazing people and gained experience with water quality research, starting from acid washing and bleaching equipment to sample collecting and sample analysis. Also, as sample collecting was very public and looked really weird with us sucking up water in a syringe and putting it in these brown bottles, there were a lot of chances to talk with the public and community members. While I didn't talk the most with community members, I was also able to observe a wide range of interactions with agencies and the community, which taught me the necessity of communicating in different ways with different people. With the sensitivity of this research, I also learned a lot about the balance between keeping information confidential and sharing pertinent information. Lastly, when I applied for PIPES this year, I did not want to do research. I didn't think that it was for me. I didn't think that it sounded exciting, and I wanted to be out in the community, talking with people, working with people. However, after this summer, I am looking at graduate programs within the UH system suggested by the graduate students that I worked with to hopefully continue working on building that pilina between science and the Hawaiian community. Some acknowledgements. I want to acknowledge my mentors, my mini mentors, the graduate students that I worked with, and the PIPES program for all of the support that they gave me throughout the summer. Mahalo nui, he mauni nao, any questions? Yeah, so.
I just wanted to know how far away was the septic system that you flushed and where you measured how long it took to get to the ocean.
Ooh. Yes. Okay, so the question was how far, sorry, how far was the location that we put the dye at from the shoreline? I actually don't remember. But I do remember that we estimated that it would take about 5 days for the dye to reach the shoreline, so probably about like 500 meters away. Yeah. Ui.
Do you plan, or how do you plan to inform the communities of that area of the sewage leaks that are going into the water?
Okay. Do I repeat this?
Okay.
So it's actually not going to be me informing the community. It is part of the graduate research that the students, Crispin and Shayla, that I was working with is doing. They have been going to the community association meetings. That is where they're planning on eventually presenting it to them. Yeah.
I'm curious about marine life. How was the areas that were tested, that you did your project, how was the marine life there? Were there a lot of life going on? Not so much?
That's the thing is we didn't really get the chance to go into the water. We were mostly on the shoreline. So I'm not really sure about the marine life in the water in that area. But what I do know is the marine life in Hilo is a lot more beautiful than the marine life on Oahu in some of the areas with sewage pollution.
Time for one more.
You mentioned about how they wanted to change all the septic or the cesspools all the way from till in 2050. All of them are going to be changed. Is like the state gonna pay for that, or are they making everyone pay for that? Like, how do they? Because that's a lot of houses, and that's a lot of like money and stuff, and a lot of change and everything. How do they expect to go about that?
Yeah, so that is actually one of the things with this research is that we're hoping to provide the results to give evidence that there is this issue, and it's a much bigger issue than just one household sewage. In order to hopefully gain funding from the government to be able to switch. Because it can cost a lot of money to switch to the sewer line or to some other alternative. Yeah.
Right on, mahalo.
Mahalo.
So yagoyangan gomwit ka Jojo. Good morning, everyone. It's so nice to see you guys this morning. My name is Sabrina Bejang.
And I'm Kara Neal.
And this summer, our project was on exploring cultivation practices for native limu at the Pacific Aquaculture and Coastal Resources Center, also known as PACRAC for short. Oh, here is an outline of our whole presentation. We will begin by introducing PACRAC, then going into giving some background information on seaweed, and then Kara will go ahead and present her project on Kalerpa, then I will present my project on Chondrus, and so forth.
So the Pacific Aquaculture and Coastal Resources Center is located here in Hilo along the Keaukaha coastline. And for short, it's PACRAC. We're going to be saying PACRAC a lot. But yes, it's near the breakwall. So the bottom right image is a picture of the location, and the top left is an aerial view of the site. So PACRAC was once a wastewater treatment center, and now is an aquaculture center since the early 2000s. They focus on cultivating fish for food and ornamental reasons, oysters, and micro and macro algae. As you can see on the top left photo is an image of tilapia, which is used for food purposes. Following that image in the middle to the right is a photo of microalgae, which is used to feed the oysters and the baby fish. Following that image to the top right is an image of Achilles tang, which is an ornamental fish. There is a lot of different species at PACRAC that are ornamental. And then following that below is an image of oysters. So there are two species of oysters at PACRAC. There are the small native Hawaiian oysters, which are used for bioremediation purposes here in Hilo Bay, Maui, and Pearl Harbor. The large oysters are edible, and they are sold to Washington for restaurants. To the left is an image of Nainui, or more commonly known as the brassy chub. This is a native fish, and they are used for food purposes. And then the last photo is an image of our experimental hale that we worked in this summer. There were two hales. This one we spent most time in just to understand the seaweed and the conditions that they really liked. So, yeah.
And because we both worked with seaweed in our projects, here are some background information. So macroalgae, most commonly known as seaweed, is considered to be a large plant-like form of algae. And in Hawaii, the Hawaiian word for seaweed is limu. And so you will see that we will mention seaweed as limu throughout this whole presentation. It can be classified into three groups. That's chlorophyta, which is the red algae, green algae, rhodophyta, that's the red algae, and phaeophyta, the brown algae. And these groups are characterized by their photosynthesis pigmentation. Limu is a marine plant that can be found in the intertidal zones. So during the low tide, you can see it on the rocky shores. in the crevices of rocks and along the sandy ocean floor. And here you can see, it shows the trophic levels. Limu is considered to be a primary producer, which means that they convert the light energy into food and nutrients for other marine herbivores such as sea turtles and herbivorous fish. Interesting facts about Limu in Hawaii is that before Western contact and to this day, Native Hawaiians regularly incorporated limu in their meals, like the Hawaiian diet, and they may have it with their raw or cooked fish, shellfish, sometimes use it as a seasoning, or serve it as like vegetable. And it is important in their culture because of their nutritional components. It was also used in their rituals, their legends, and or social activities. Now the picture on top you see here is delicious ahi poke with limu. And then on the bottom it shows spam musubi, also wrapped in seaweed, and both of which are staple food in Hawaii today. And now that we have learned some basic background information about seaweed, let's give Carol our attention as she presents her summer project.
So the seaweed I focused on this summer was Caulerpa lentilifera, and my project focused on improving cultivation practices using fish effluent. Caulerpa lentilifera is more commonly known as sea grapes. They are favored due to their grassy green color and texture. This seaweed grows in nearshore waters on sand or rock bottoms. As you can see in the image on the slide with the arrow pointing, that's the area where you can find the sea grapes. And they are also very valuable for human health like other seaweeds. They improve or increase the intake of dietary fiber, lower chronic diseases, as some studies have stated, and they are brain food. So the current system right now at PACRAC for cultivating sea grapes is flowing seawater with a constant temperature of 23 degrees Celsius, fed nitrates, covered in shade cloth, and are provided air stones for aeration. We know that the Caulerpa likes these conditions because they prefer very little sunlight, warm water, and nutrient-rich water. So since they're doing so well, as you can see in the image, they're thriving, and we were like, so what's another way that we can grow Caulerpa? So this led to my research question. How does Caulerpa respond to an integrated system with fish effluent? So for my study, again, I focused on how to improve cultivation practices using fish effluent. On the left picture, you can see that this is an oyster cage, which is 3 feet long. We put the Caulerpa into 6 oyster cages. 600 grams were placed in each cage. There was 3 tilapia tanks with flowing seawater at an average temperature of 20 degrees Celsius and air diffusers. 2 cages were placed in each of the tanks on opposite sides of one another. Each tank contained a different life stage of fish. There's juvenile, teen, and adult. On the right-hand side, you can see one of the oyster cages submerged in the adult tilapia tank. All oyster cages were labeled so we could keep track of which. Water quality parameters were also taken so we can keep track of the pH, salinity, dissolved oxygen, and the water temperature. We also took the weight and light measurements once a week. So these data helped also answer our research question, which analyzed Caulerpa's growth across 3 integrated systems, which differed by fish age. So the first graph here as you can see on the y-axis, we have our growth rate percent per day, and on our x-axis, we have the fish sizes, the juvenile, teen, and adult. And the blue bar represents week 1, the orange represents week 2, and the gray represents week 3. The main finding from this graph is that juveniles are bad. They were found to have a significantly negative impact on the caulerpa's growth rate. This next graph is showing the growth rates of caulerpa over time. The x-axis represents time, grouped by weeks, and the y-axis, again, we have growth rate percent by day. In contrast to graph appearance, our statistical analysis indicated that there was no difference in growth rate over time. We suggest using more replicates for future studies. So on this graph, so where are the differences? After we did a statistical analysis in R, it showed that juvenile had a negative impact on the caulerpa growth, while the teen had a positive impact on change in weight. And then the final graph, patience is key. So the first day, the growth wasn't looking so great. It was starting off really slow. We were starting getting worried, like, oh no. But by the end of week 3, it was thriving. It was growing very well. So this shows that caulerpa can improve over time. The longer we would have kept it in there, the more chances it would have had to grow. So what does this all mean? Sea grapes do not grow well in the juvenile tanks, therefore suggest to experiment with teen and adult tanks in future studies. The more time the species is given, the more it thrives, which suggests cultivation practices will be a longer time period. By week 3, again, they were looking great and fantastic as they loved the fish effluent. From this study, the sea grapes continue to stay in the tilapia tanks to continue to observe the healthiness and firmness of the species. We actually just checked it 2 days ago, and they're looking a lot better than in this image. They're looking a lot darker and firm. So I will now give this time to Bree to present her summer project. Thank you.
Thank you, Carol, for sharing with us your awesome project. Hi, it's me again. For my project, I wanted to know what are the effects of salinity on the growth and photosynthesis of Hawaiian chondrus species. Chondrus, which is in the rhodophyta red algae group, can be found in temperate climates in coastal areas of Korea, Japan, Chile, Canada, and Hawaii, just to name a few. It is considered to be an economically important seaweed because of its carrageenan content. Now, carrageenan is just a natural compound that is extracted from red edible seaweeds, for example, chondrus. Products containing carrageenan content are all around you. You can, it is in ice cream, chocolate, yogurt, baby formula, pet canned food, even in your toothpaste, your lotions, and some makeup products. And because Hawaiian chondrus, studies on Hawaiian chondrus are limited and with an increased demand in the food and biochemical productions in Hawaii, it is important to find ways to improve its growth rate. And one way to do that would be to learn how to grow it in controlled environments. And for this project, we grew chondrus in various salinity treatments by manipulating the salinity levels. And the question is there a significant difference in growth and photosynthesis efficiency of chondrus grown in 3 different salinity treatments of 11, 15, and 31? Thank you. And to set up the experiment, 9 grams of chondrus were placed in a 2-gallon Ziploc bags that each contained one of the 3 salinity treatments, 11, 15, and 31. Now, each Ziploc bag was then placed in a 5-gallon bucket that had flowing seawater in it at 20 degrees Celsius. An air stone was placed into each bucket to regulate aeration, and each bag was secured shut and supported upright with the bucket with clothespin. So you see here, the first picture, it shows what each bucket looked like on the inside. And this bucket, this picture on the right, that shows all 9 replicas. So for each of the 3 salinity treatments, there's 3 replicates of these buckets, so making 9 buckets in total. And you can see here that they're all under the same shade cloth so that the conjures does not get too much sunlight. And they're all receiving the same flowing seawater at pack rack to make sure that they all had the same temperature, which was at 20 degrees Celsius. And once every week, a YSI was used to check water quality and to monitor the salinity levels to make sure that each bucket was still set at its desired salinity level. A pulse amplitude modulation fluorometer, also known as the PAM meter, was used to measure its photosynthesis efficiency. Now, photosynthesis efficiency just means that we're monitoring like how well it is photosynthesizing, like changing, converting the light energy into nutrients or energy. And for each week, chondrus was fertilized with half a teaspoon of Miracle-Gro, and then all the data was saved and recorded in Excel. And after the collection, after 2 weeks, we analyzed the data using non-parametric and parametric tests in RStudio software. So here is a graph that's showing the growth rates of chondrus across all 3 salinity treatments of 11, 15, and 31. On the x-axis, we have salinity. The y-axis shows the growth rate as percent per day. Now the blue bars represent week 1, and the orange bars represent week 2. And the data analysis indicates that growth rates were similar across all 3 salinity treatments, but we do see a trend here. So on week 1, we can see that the chondrus grown in the 15s and 31s did way better in the ones grown in the salinity levels of 11. And on week 2, we can see that chondrus grown in the salinity of 31 did not grow as well as the ones grown in the other 2 salinities. And here is a graph showing growth rates over time. On the x-axis, we have week 1 and week 2. Y-axis shows growth rates as percent per day. We can see from week 1 compared to week 2 that chondrus growth rates decreased significantly. And on the third to last slide here, this graph shows photosynthesis efficiency across the salinity treatments of 11, 15, and 31. On the x-axis, we have salinity grouped by treatment. The y-axis, we have maximum quantum yield, which again represents photosynthesis efficiency. The blue bars represent week 1, and the orange bars represent week 2. We can see here that the photosynthesis efficiency is similar across all salinity treatments. And the last graph is showing the photosynthesis efficiency over time. On the x-axis, we see week 1 and week 2, and y-axis, we have the maximum quantum yield. And again, we see that the photosynthesis efficiency seems to stay similar over time. So what are our findings? Our main finding here is that chondrus growth rates were decreasing over time while the photosynthesis efficiency was similar or keeping up. Growth rates decreasing over time suggests that our systems may need improvement. So like maybe more replicates would have given us more data. And despite chondrus losing weight over time, photosynthesis was seen to be consistent throughout the whole experiment. And these results suggest that The species was consistently generating energy, yet that energy was not being used for growth. And fortunately, we don't know what it was used for, but we hypothesize that the energy was used to like mitigate stress. Therefore, more studies are needed. And that concludes my presentation on chondrus. And here's some pictures that we picked out to kind of show what our summer was like. The first picture on the left over there is me and Kara, and that's what a day in the Hale looks like. Cleaning limu buckets, cleaning it from all the diatoms that has grown over time. And my favorite picture here, that's me and Kara with our other mentor, Rose Cristiane. That's the day we learned how to bake, incorporate limu in cupcakes. And we baked it for our last huaka'i.
And the picture on the right is the picture of the dog at Pack Rack. She is the guard dog, the princess. She is so sweet. She'll make your day a lot better. And then also one of my favorite photos is on the bottom, is when we went to Kalapana tide pools to help fellow interns gather seaweed and identify with Dr. Carla McDermott.
With that being said, we would like to thank the National Science Foundation, Hau'oli Maoloa Foundation, and the Pacific Internships Program for exploring science for the wonderful opportunity to be a part of this program.
We would also like to thank the University of Hawaii and Pack Rack, and of course our mentors, Dr. Carla McDermott and her grad student, Rose Cristiane, for their awesome mentoring, guidance, and support.
And we would like to thank all of you for being here and you tuning in, for showing your support. Thank you. If you have any So if you have any questions, please don't hesitate to ask, and we'll try not to hesitate to answer.
Yeah, I have a question for Sabrina. How did you choose the salinity levels for your experiment?
Oh, that's a good question. I read in a paper, I can show you later, that chondrus can be grown, like, it's resilient in salinities as low as 11. And then the salinity level for the flowing seawater at Pack Rack is actually at 31, I believe. So I chose, I started with 31, and I was like, oh, what if we like half that? And half of that is like about 15, and the lowest that I read about was 11. So that's why we chose those numbers. So it's like full salinity, half salinity, and low salinity.
Thank you. I have a question for Kara. Do you know why the growth rate decreased in the juvenile tank? Were they like eating it, or?
So yeah, so I think there is two reasons of why it wasn't doing great. The juveniles are actually able to fit into the oyster cages, so they were being a pain like moving around the caulerpa. And another reason is that there was, I think, 200 or 300 juveniles in the tank, so I think it was excess like nutrients. So it was probably like just too much nutrients for the caulerpa.
Great. We have a question online from Joe Gentz. Thank you, Kara and Sabrina, for a great joint presentation. Sabrina, are you aware of any similar cultivation practices / uses of sea grapes, chondrus, or other types of seaweed in the Marshall Islands?
Oh, thank you, Joe Gens, for your question. That is something I will look into. Unfortunately, I don't know if there's any similar ways of cultivating it in here in Marshall Islands, but I will look into it and get back to you. I know Joe Gens.
I just had one follow-up question too. Is chondrus a native species?
Oh, yes. chondrus is indeed a native species. It can be found, you can see it in like Alon Richardson's and Violena Beach.
Cool.
Yeah.
Mahalo.
Thank you.
What is the Hawaiian name?
For which one?
There's actually
For both.
no Hawaiian names for these two species because they weren't really used in history. Yeah, so they're just now getting get to know the species more and how to use it. Yeah.
There's we were talking about this. There's a lot of Hawaiian names, like a lot of other species that has Hawaiian names, but chondrus and Caulerpa just.
Yeah.
Maybe you guys can name them.
Ooh.
Good morning, everybody. My name is Cole Dilde-Sa, and I represent a number of people working at the USGS, UH Hilo, and the state of Hawaii's co-op lab. And our project, or one half of our project, which I'll be presenting today, is using social media to understand COVID-19's impacts on Hawaii Island's fisheries. So, an introduction. So, we all know what COVID is. It's caused a massive shift in the way we live our lives. We can see it here today for everyone sitting in the building. And you know, we know that there's been a massive change in how we live, but we don't have a lot of hard data to support how exactly and in very specific ways that COVID has impacted us. So this is sort of the crux of what we were doing this summer. Our project aims to understand how COVID impacted Hawaii Island's fisheries. And understanding how COVID impacted the fisheries will help us better manage our response to traumatic events such as COVID-19 in the future. And especially other traumatic events caused, thinking in the future, towards the future, caused by climate change, for example. Like how do people react to large disturbances in the social economy? And so there are a couple problems with our project in specific, or our project in particular. There's not much information collected about the island's catch statistics. So Hawaii is one of the actually very few states that doesn't have a registration process or mandatory reporting of catch for fish. And another problem is that it's hard to collect this data. You actually have to go out, talk to the people on the rocks, at the beach, whatever, and ask them about their fishing. So how do we get around this problem? Social media. So we've all seen that guy or that uncle, that auntie with the fish, big fish right there. And people like to talk about fishing. They like to post it on the internet, and the internet and social media can be used as a source of a massive amount of information that we can analyze to understand our catch in Hawaii and in the world at large. It's an established process, but only on the mainland. It hasn't been tried out or hasn't been put to the same effect in a diverse fishery like Hawaii Island. And so, yeah, instead of going out and collecting the data, we decided to let the fishermen provide it to us. So another implication is that understanding this process and applying it in such a way that we have here will allow us to more effectively understand the fisheries in the future and allow the government to properly respond to, again, COVID-19 traumatic events like that. So we have 3 questions for our presentation today, and 3 questions that sort of guided our research. Are people fishing for more fish? And this is in response to the pandemic. Are people fishing for an increased variety of fish? This has to do with how diverse people's catches are per post or per trip. And third question is, are people fishing for different types of fish? Are they targeting something else in response to the pandemic? So how has catch composition changed between pre-pandemic and pandemic periods? So our methods. Our study site is, we took posts on Hawai'i Island. We used Instagram's features, which make it very easy to filter down to site-specific places, like hashtags and geotags, and we use post context to help filter this data, and I'll show you an example, a couple examples on the next slide. We narrowed a study site down to posts taken between the years 2016 and 2021, and we used the date March 23rd, which is the first state-mandated lockdown issued by Governor Ige, to delineate the pre-pandemic from the pandemic periods. A little bit about data collection is we basically went through all these posts, recorded number of data points, date, species, count per species, gear type. So, next slide. Here's a couple of example posts that we might have used. And so you can see, remember that uncle? See him on the right? But, so, we would look at posts like this, basically scrolling through Instagram. We see on the left where there's an omilu. Whoever posted it is used a fishing pole. There's one omilu. We would do like count. And so we would just use contextual evidence, sort of trying to break it down and see, if you can see on the left, it says @BigIslandBaits, and then under that is like hashtag, on the top, it says hashtag Big Island Fishing. So that's how we would know it's site specific and date specific. I don't think any of the dates here, but it's just a little bit. It's on Instagram. And so, this is just what we were working with. So, methods, data analysis, how did we analyze this data? Because it's very important to understand the hand-wavy stats behind all of this and to draw statistically meaningful conclusions. So we used a zero-inflated mixed model, a Poisson mixed model, to analyze our data. And this just means zero-inflated describes the distribution of our catch count and our species count that we got from our data, and I'll show that on the next slide. And a mixed model has to do with the effects that we're dealing with within the model. Really all we need to know is that it's mixed with fixed and random effects. And so fixed effects are sort of the effects that we're working with, and then random effects are things that we have to take into account because of the nature of what we're working with. For example, years before 2016, right? We didn't collect any data for that. It might have an impact on sort of what we're dealing with. But here's our distribution. So on the left is our data that we were working with. As you can see, a majority of the data set, 65% about, we were dealing with, 65% of the data count comprised of one fish per one post. And then it, tails off in the end. And see on the right is a Poisson distribution. That's how we matched it. So moving on to our results. Okay, so this actually wasn't included in the model. This is just sort of preliminary data. More posts were posted during the pandemic period. See on the right is the average amount of posts per month for pandemic period. This is March 23rd, 2020 onwards. And then pre-pandemic period is March 20th, 23rd previously to January 1st, 2016. And so we can see that about twice the amount of posts were posted during the pandemic period, sort of indicating that people were posting more. Next one. So in reference to our first question, are people fishing for more? Yes, more fish were caught during the pandemic period according to our Instagram data set. About 25% more, in fact. And the story behind this, what's the story? We think that the difference is caused by the COVID-19 pandemic, most notably because of job loss, people having more time to go out, cast net, throw net, go spear, go cast their line out, and basically more people trying to catch more fish. And so, moving on, we have the average amount of species per post. This is our second question. The number of species per post did not change between pre-pandemic and pandemic periods. What's the story? Maybe it's more efficient just to go for one species per one time out. But yeah, there's no statistically meaningful difference between the amount of species per post. Our third figure is dealing with the third question, the species composition between the periods. So species composition changed between pre-pandemic and pandemic periods. People were fishing for different fish, and as you can see on the left, we have ono, we have mahi-mahi, and ahi. Those are the pelagic fish that we were dealing with, and they were fished for heavily in pre-pandemic compared to pandemic periods. And then we have coastal pelagic, that is ulua and omilu, and we can see that they're fished for heavily during the pandemic period. So we think this has a lot to do with the boat restrictions that were put into place during the pandemic lockdowns. So people moving from boats onto the shore. So our discussion, what are our main takeaways? People caught more fish, people did not catch a wider variety of fish, but the types of fish that people caught changed. And moving on, our assumptions that we have to take into account is that Instagram users equal a representative amount of the fishery as a whole. So Instagram users represent the fishery. We also have to understand that one of our assumptions is people photograph their entire catch. And another interesting point that we have to say is people don't report their zero catch trips, so we have no idea the amount of effort that people are putting into fishing. And then lastly, sort of going back to the broad general essence of the presentation is, although this is sort of a rudimentary application of like social media analysis, it has a lot of potential. And more specifically, it has a lot of potential to be used to help people. And so our research suggests that these methods can be used more effectively to manage our natural resources in the future. So thank you to PIPES and all of our funders. And thank you to Tim and Michelle and other Michelle at the Hawaii Cooperative Fishery Research Unit for mentoring me this summer. And any questions?
I know you said in your assumptions that you were assuming like the Instagram users were the representative of the whole fishery, but do you have like an estimate of like how many, like what % of the fish that are actually caught are being posted on Instagram, or like what that would scale to?
No, I think the only thing that I can say to that question is that I mentioned earlier, but I'll reiterate that there's been literature pointing, like scientific papers published in like natural, like some fishery management reserves in Montana or somewhere in the Midwest, basically using social media, saying that, and proving the effectiveness of social media as a good data collection tool. And so I don't know exactly like how many fish are on social media compared to the, we didn't test for that, but the fishery in general, but we didn't test for that, so. No.
I know obviously that your project, the emphasis was on social media, but were you able to conduct any like in-person physical question?
That's a perfect question, actually. So this is, I mentioned one half of a two-part project. My brother man Drew is gonna come up here and explain the other half of what we did and sort of the broader implications that both of our projects coming together point to.
Yeah, mahalo for that. Cool.
Thank you.
There was a slight change up. So on the schedule, it said that Drew Curley was going to be presenting, but we changed it up a bit because by request. So our next guest speaker is going to be Drew Curley.
All right. Hello, everyone. So like Cole said, my project is kind of tied in with his. It's just kind of the in-person aspect of that online research. So whereas Cole's project, we went and did research on Instagram, here we went and talked to uncles like this guy just fishing at Breakwall. So the title is Responsive Hawaii's Nearshore Fishers to the Impacts of COVID-19. I worked with Cole, Michelle Shui, Tim Grabowski, and then Michelle Benedum. Down is forward, okay. All right, so we had one overarching research question. It was, how has COVID-19 impacted the fishing activity, livelihoods, and practices of nearshore fishers in Hawaii? And so to answer that, we had all these questions beneath it. We had, how was your work frequency impacted by COVID? How was your effort? Like, how often did you go fishing post-COVID? What was your motivation? Food, fun, both? How often do you fish just in general? Do you rely on your catch as a part of your diet now more than pre-COVID? Has your catch changed since COVID? And then have you started targeting different species? So just a little overview of the impacts of COVID in Hawaii. To delineate the start of the lockdown, March 23rd, 2020, like Cole had said, that's when IGE enacted the first stay-at-home lockdown order. And then after that first one passed and everyone was allowed to, go out into the world a little bit again, some of the allowed activities are fishing, surfing, running, stuff you could do outside away from people. They allow people to get out of the house, get away from family, you love them, but sometimes they're a lot. You go surf, clear your mind. And then a lot of people lost jobs and had surplus free time due to lockdowns, restrictions, and you couldn't do much, so you go outside. Little overview of the Hilo Bay fishery. It's typical of most nearshore fisheries in Hawaii. It's largely non-commercial, a lot of guys just fishing for fun or trying to get some food for friends and family. And then commonly targeted fish, ama, the mullet, ahole, Hawaiian flagtail, halalu is the young big-eye scad, young akule, nanui, uhu, all those reef fishes, and you guys have seen them. So methods, we use access point surveys during July 2021. We had 15 sites where, I'll have a bigger map in a couple so you can see all the sites, but from Bayfront to Kings Landing, so just one half of Hilo Bay really, the other half is kind of hard to access. We had a total of 69 anglers that we talked to, and we had 17 discussion prompts, just kind of open-ended and yes or no guided questions to keep the conversation on track. And these topics were generally like those research questions we had. You fishing more or less? Are you catching more or less? And then we had a comment section just for stuff they've noticed. And these comments really helped us kind of break down the answers and explain why some of these things are happening. And then no demographic or personal info was collected just to assure anonymity. And then characteristics were noted like, oh, so-and-so, like this much females, this much males, older, younger, just stuff like that. So for data analysis, we converted, a lot of our stuff was verbal, like yes, no, or stories, and we turned that into numerical. So like a yes was a 1 in the spreadsheet, we put it on, no, -1, same, 0, stuff like that. And we put it into SAS, statistical analysis software. And through this, we use chi-squared tests to look for significant trends in the data. And then, like I said, that comment section, once we got those results, we saw the trends, like maybe this was happening because so many uncles said, yeah, I saw way more families fishing at Wailoa, or I saw this, I saw that. So the comments were really helpful. Results, just some of our basic observations. Site population really went up with ease of access. So like Mokuola, Coconut Island, Bayfront, and Wailoa River State Park, those three give us 46% of our data. And that's where there was a lot of older retired uncles and guys who just wanted to be able to drive up to their spot. And then some of the characteristics of these fishers, for age, a lot of the older guys fell into our both category, which was I fish for fun and food. And then a lot of them fished less, just concerns about COVID and they wanted to stay home and not risk it. And then gender, about 1 in 10 anglers were women. There's no difference in their results. Everyone basically said the same thing. Well, not really, but there was no crazy difference there. And then gear type, it was a lot easier to talk to pole fishers because guys who are casting net, they're in hunt mode, they're stalking the reef, and you're not going to go wait out to uncle and he's trying to hunt. You're like, excuse me, sir, can I talk to you? And then guys who are diving, 95% of their time in the water. So that's why like drastically more of our data came from pole fishers because you can walk up to them. This is the map. So these are our study sites, and those 3 in red is where the 46% of data came from. But yeah, 15 sites stretching along the coast there. I'm sure a lot of people in this room have been to most of them. So now we're getting into our results, our graphs and stuff. So this first one shows that subsistence fishers had a greater change in work hours. So on the x-axis, we have their motivation fun, both food, recreational, both subsistence. And we can see that subsistence fishers seem to have, I don't want to say more volatile, but their work was more shifty. They're working more, they're working less. And that was just an interesting thing we found. And that leads into fishers who worked less, fished more. And this one, huge trend. You can see there on the left, about 90% of those who decreased work increased their fishing. And then same on the opposite end of the spectrum, those who increased fishing or increased work fished a lot less as well. And then this one is a box and whisker plot. The red lines are the means. So we're looking at a fisher's motivation compared to their average trips per day. And you can see that goes up from recreational about 0.4 trips per day, subsistence to about 0.55, 0.6. And the way we got that was we would ask people like, oh, how much do you fish? And oh, 3 times a week. So we take that 3 times by 56 for every week in a year. We get that number for annual trips, divide by 365, and you get your decimal point there. And then calculations, subsistence fishers on average took about 73 more trips per year than recreational fishers. And then fishing frequency compared to motivation kind of split the data set. It showed who really needed the food more or who was there more just for fun. So you can see on the subsistence side, a lot more guys were fishing more, and then recreational guys are fishing less. But in that both category, you see that even split of who was fishing more for food in that both response and who was fishing more for fun. So we thought that was pretty interesting. Subsistence fishers relied even more on their catch. So a lot of these findings are showing that subsistence fishers really were hit pretty hard by this pandemic. And half of subsistence fishers responded that they actually relied more on their catch now than before COVID. And none in the recreation, because they're just out there for fun, recreation. And both category, about 25% said that they relied more on their catch. And then these subsistence fishers kept more of what they caught, which would make sense, because if you're fishing, if you're relying more heavy on your catch, you're going to keep more of what you catch. So that kind of reflected the same as this last one. You got a big bump at subsistence and then a littler bump with the both category. And these, wrong way. The subsistence fishers even targeted new species. So that's about 40% of them branching out to new stuff. A lot of these guys were saying, oh, I'm trying to get away from people, or there's too many people fishing these grounds, there's not enough fish, so I'm going to like spots I know have fish. And in the recreation, that was a handful, maybe 1, 2 guys. Same thing, we think those are guys who just want to get away from people, fish different spots, but not in pursuit of food or anything like that for the recreational. Concluding, COVID had significant impacts on Hawaii's nearshore fishers, and fishing in total has increased due to lockdowns, their effects. People have more time, less work, go out and fish, catch some food. And then, like I said earlier, subsistence fishers seem to be hit the hardest, and this came in the form of increasing their frequency. They relied more on their catch as part of their diet, they increased the amount of fish they kept, and then they targeted different species. And then, so this is where I'm going to compare my research to Cole's, because although it's great to go out and talk with uncles, that's kind of hard, especially in COVID. So that was like a big concern, and that's why the research wasn't done earlier, because you weren't allowed to approach people. So there's a very large difference in effort. Whereas when Cole and I were on Instagram, we could sit down, we could grind out the 69 responses we got for this in a month. We could do that in a day. So that's why Cole's data set was something like 350 individual posts or pictures. So that could have been 350 guys, whereas mine was 69, and it took way more time. So another interesting thing, the results actually pretty similar. And for the amount of effort that was different in those, that's alarming. And that begs the question, can social media research be a viable substitute for traditional research? I think that social media research needs some more testing and more data to see how close we can get these results to be and if it is accurate. But one thing that you're never going to get with social media is the nuances when you're talking to someone and they say like just little stuff, like the amount of times that guys would throw in some cool history about this or just a little something that you remember when you're looking at data and you're like, oh, Maybe that's why you can't get that from Instagram. That's just, like you saw in my picture of Cole, it's just, there's the fish. So that's something that you can't get from online. And then there's different biases for the two types of research. With online, you're kind of looking more at a population that is younger and more tech savvy, because that's the majority of Instagram users. And then with traditional research, just going face to face, talking with guys, your sample size is harder to build. You can't talk to people under 18 just for permitting and all that, all those reasons. And then you underrepresent the divers and castnators because they spend a majority of their time hunting in the water, not able to be bothered. Like a couple times we actually walked up to castneters and it didn't look like they were doing much. Like we see them loading up their net and we're like, oh crap, he's about to throw, we gotta go. And so we walk up, oh, can we talk to you? Oh, I'm catching bait right now, sorry, can't. It's like, okay. So we miss out on those, but on Instagram, guys get their loaded nets and you see what they get and all that. And then not everyone wants to talk when you approach them. Like certain sites, I'm not gonna call any sites out, but certain sites, there's not very friendly people. You got turned down like 3 out of 4 times and just, okay, have a great day. Yeah. And that wraps up my presentation. Mahalo everybody here and there. I'd like to thank PIPES, National Science Foundation, University of Hawaii at Hilo, Hawaii Cooperative Fisheries Unit. And that fisheries unit there's a little blurb down there. It's a bunch of acronyms and peoples working together for science. Yeah. Thank you, everybody.
Did you guys consider reaching out to the people you saw posting on social media, like directly messaging them to ask them similar questions to how you surveyed them in person?
No, that's a great idea, though. That could be a way to get those nuances and the subtleties of like conversation and some more background info on the fish. It's nice, yeah. Should have thought of that.
Kind of similarly, did you guys, I think we talked about this at one point, did you guys approach tokunagas to see like if there had been a shift in any way in gear, that kind of thing sold during the pandemic, since the pandemic started?
So we didn't approach tokunagas directly, but a lot of fishermen would say tokunagas is out. They don't have baits, they don't have lures, they don't have this, they're out of 3-prongs. And I tried to buy a 3-prong during the pandemic and they didn't have what I wanted, so I couldn't get that. So definitely was, and I heard this is like universal across all islands. There was no tackle, you couldn't get it shipped in. Bodyboards, surfboards, everything. All ocean gear, because that's all stuff you could use recreationally. People were like, well, I got nothing to do, let's go buy some fishing stuff, let's buy some, yeah. But yeah, we did not directly approach them.
I actually have a quick question for you, Drew, and maybe Cole can also fill in as well. But I was just wondering if you noticed any like difference in size, maybe like if people are fishing for smaller fish now that they're maybe perhaps getting a little more desperate for food or subsistence. Did you notice any differences in size or anything?
So that wasn't a question that we asked, but I think even if we did, guys wouldn't say, yeah, I'm keeping undersized papillos all day. Like, no one's gonna come out and say that. But I don't know, a lot of guys are fishing, like I saw people take to'aus, but I mean, there's no size limit on to'aus, it's invasive. To'aus are maybe 5-6 inches long. But no, we didn't, no significant findings for size. Yeah. Thank you, guys.
Mahalo, Drew, for that. We have our next presenter, Jessica DeCosta, who will be talking about her experience this summer. Mahalo.
All right. Hello, everyone. How are we doing? Almost lunchtime, huh? Alrighty. So, oh, what you doing to me? Raj. So, aloha mai kako. My name is Jessica DeCosta. I am originally from Kaneohe, Oahu, and currently living in Hakala, and a UH Hilo student, also a PIPES 2021 summer intern this year. So my presentation is called, today is going to be a Winter River Jumps, a peek into Waipi'o. I worked this summer at a traditional lo'i taro patch restoration site called Kapapa Lo'i o Keali'i Kua'aina. I am highly honored and very blessed to have been paired with an awesome mentor, I'ini Maikalani Kahakala, or I'ini for short, this summer. Someone who has many hats, many titles, but also has roots very deep in Waipi'o that trace back many generations. So, a little background on Waipi'o for you real quick, or maybe I'll kind of divulge a lot, because Waipi'o is very rich of history, and very rich of community and culture and everything that goes down there. So Waipi'o is located right outside Honoka'a town, right along the Hamakua coast on Hawai'i Island, a valley that has always been very fruitful, and a long history of communities in Ohana finding homes down in Waipi'o. Well known throughout time for its ability to cultivate an abundance of year-round harvests such as kalo or taro, and fish from the local i'a, or fish ponds, like lalakea. Since the plantation and ranching days, Waipi'o has seen rice, mai'a or bananas, ko or sugarcane, mac nuts, and some citrus trees, just to name a few. The valley always having been able to provide and produce for populations and communities up to the thousands for generations. In Hawaiian culture, the kalo is a sacred plant that speaks to the relationship between the man, aina, and the natural world. Kalo can grow almost anywhere. You hike all over Hawaii, you're gonna find them growing in the waterfalls in the gulches, you'll find them growing on the banks, you'll find them growing everywhere randomly. It is very a hardy, staple kind of a plant, and it survives. It definitely kumakani. Kalo can grow in dry land or wetland. It grows primarily from cuttings or shoots, keiki cuttings or shoots from the corm. And it also prefers cool waters or colder waters. It doesn't like the warm waters to grow in as well. And it can take about 9 months to even like over a year to harvest. But you can pretty much eat the whole thing as well, or utilize the entire plant. So horses and donkeys were once the main way of transport in and out of the valley. Since then, wild horses like this one roam freely in the valley. This is a little baby one, this one, a little patched baby one here. These wild horse populations are a result of unwanted breeding amongst the herds of farmers and ranchers. Just like any other species in Hawaii, they thrived on the abundance of food and lack of predators. They roam the beaches and the valley with their young, eating all the native vegetation because they love it. And especially like the ohia'i, all the mountain apple, all gone. No sense in grabbing, they got them. Eating all the native vegetation and farm goods that they can access. They also have to be aware that people who live down there, they gotta plant their plants like 10 feet from their fence, otherwise the horse is gonna reach over and go eat them all. Actually preventing the horses from breaching the fence was also kind of part of my job. You never know what you're gonna say to a horse to get it out. These just weird noises come out. Anyways, one of the best ways to kind of, like, when you Google Waipi'o, you're not going to get very much information at all. You kind of have to, like, kind of dig, and talk to the community members to really know what happened there. But one of the best ways to kind of understand Waipi'o is through mo'olelo, or Hawaiian mythology, Kalamai. From the Hawaii Island legends compiled by Mary Kravena Pukui, this one is a quick mo'olelo or shortened version of the story of Kila, the youngest son of High Chief of Mo'ikeha and High Chiefess Ho'oipoi Kamalanai. I practiced that so many times. Her name is hard. Kalamai. Ho'oipoi Kamalanai from Kapa'a Kauai. When Mo'ikeha dies and leaves Kila, the youngest son, as chief, his other older brothers become super jealous because he's granted it. He was the baby. They devise a plan where they all travel by canoe on a voyage for their recently passed away father, only to abandon Kila while asleep in Waipi'o. There he works as a servant, only to become hanai'd by the ali'i, the chief that's there, because he sees that he actually is a chief as well, and he hanai's him and makes him, here, you can be chief as well too, and he takes up the role and cares for Waipi'o Valley as his own. unfortunately, Waipio wasn't doing very good at the time. He was tired of seeing his people hungry. They weren't getting, they were only harvesting small kind and then they're not getting year-long harvest. So he changed it up. He created a farming system and he made it where everyone has kuleana over their own kalo patch while taking turns working the chief's one. And with great success, it generated the abundance and stability of Waipio so greatly. They had so much food, word got out to the other islands. Kila's ohana back in Kauai heard about it, and they actually didn't have enough food to feed their people at the time. They were actually kind of starving. So Kila's brothers, unknowingly that he was chief now, they went over there, and now them, chiefs of Kauai, went over to Waipio to go get some food for their people, only realizing that Kila's now chief, and they're like, oh, now he's still there, he's alive, he's mad at us because we abandoned him, right? So after some conflict and him being like, all right, I'm gonna teach you a lesson a little bit, he goes ahead and gives his family food and sends it back to Kauai. However, he stays in Waipio and lives on as chief. As a happy, happy chief, actually. His people were very stoked. This mo'olelo, I chose this mo'olelo because it's so revealing how bountiful and giving the land can be, especially when you work as a community. Because that's one word that comes to mind when you think of Waipio is community. Okay, Waipio has suffered, Waipio is known for its beautiful tall mountains that hug the valley with cascading waterfalls like Hi'ilave, one of Waipio's 5 main sources of water that feeds into the rivers, flooding especially when it rains. The valley attracts hundreds to thousands of visitors and locals every day to hike down from Waipio Valley Lookout to the black sand beaches that also draw in the local surfers, even though it's limited access with one small road requiring 4-wheel drive. Waipio has also suffered multiple floods throughout its history and tsunamis, like the tsunami that devastated Hawaii Island in 1946, destroying lo'i farms, homes, and changing the whole topography, causing rivers to jump or redirect in Waipio. Many lost homes were forced to relocate. Many lost homes and families were forced to relocate out of the valley. Today, Waipio is still producing pounds of kalo and is home to a close community where everyone knows one another and waves. Every time they pass by, they wave to each other, and it's really awesome. So I kind of want to point out too a little bit. This is a good photo because you can kind of see the tsunami stuff. It kind of pushed back this far. It pushed all the sand and silt and pushed it kind of far back. And so it kind of destroyed a lot of the land. So it was kind of fallow after, and you couldn't really farm in it. So the tsunamis really kind of destroyed the land too. So it made it harder for the farmers. So that's another challenge that they have as well too at this lo'i. Okay. Sorry, what are you trying to figure out this button? Okay. By the way, this is Hi'ilave right here, if you can see it behind the clouds. And then this is Kunaka here. Anyways, down in beautiful Waipio Valley, in the Ili'aina, or land area of the Ahupua'a, or land division of Kunaka, is the Rikindo Kapapa Lo'i o Keali'ikua'aina. The lo'i covers about 5 acres of wetland kalo, or taro patch farm, with one patch already harvesting from, and they have plans for about 10 more patches. There are roughly 3 acres of farmable land on this plot, due to 2 acres of being like roads, sections, and then river flows and stuff like that too. The landowner is Bishop Museum, who also owns about 80% of the valley as well too. Okay. Kapapa Lo'i o Keali'ikua'aina is cared for by the oh my gosh, all these Hawaiian words, Kahakalau Ohana, by their cultural practitioners and educators. They are also part of the Kua Kanaka organization that strive to revitalize Hawaiian language, culture, and traditions, restore Hawaiian traditions, and reconnect us with Hawaiian heritage. With plans to cultivate 10 taro patches of multiple Hawaiian kalo varieties like polulu, and pokikea, for example. They are so motivated to grow for ohana and cultural purposes, as well as demonstrating sustainability through Hawaiian traditions and practices, emphasizing the importance of Hawaiian self-sufficiency through cultivating the land and growing food. They utilize the lo'i as an outdoor learning laboratory for eco-versity learners, DOE, and private schools, cultural practitioners, and others who are interested in learning cultural-based taro cultivation of all ages.
When first granted the land, it was completely overgrown and overrun. It had been untouched for about 30 years or so. So this bottom picture right here is kind of a small photo of like what they were dealing with. And then it kind of reverts to like over here, it clears up a little bit more, and then here's what you have kind of more today kind of a stuff. And so it took some heavy machinery and effort to reveal the lo'i that was underneath it, because there is still the lo'i still there. All the formations, all the patches still there, just overgrown. And so basically my tasks this summer were to basically help them in cleaning out more vegetation, preventing further growth, invasives removal, as well as the horses as well too, and vegetation removal, weeding, maintaining the lo'i, volunteer coordinating, planting. I started this kind of a weed management project utilizing cost-effective vinegar-based natural herbicides that due to user error and some time issues, it did not really work out very well. So some of the threats and invasives that they have down there, that's kind of a problem as well, and a growing problem is the range of vegetation from animal to human problems they have. So first one would be like the apple snails, the top right corner there. So that's like apple snail there, they're freshwater snails, they're more native to South America, and were brought here to be farmed initially and sold as like escargot, but things happened and the farmers kind of there was like a flood again too, and they all kind of spilled out into the rivers and then were accidentally released, and now they freely flow throughout the rivers, causing damage to the kalo and other plants by eating young shoots or chewing into the corm, allowing bacteria inside and ultimately damaging the corm, which is the bottom of the kalo. They have bright pink eggs that are often found on the stalks of plants or the kalo and are also known to carry lungworms, so you really don't want them in your kalo or in your patch. Years ago, another problem would be fire ants there. They have, of course, Hawai'i got introduced to fire ants many times ago, and so they quickly spread at rapid rates. First clearing of the lo'i, I was told that it was greatly infested. You couldn't even sit down on the ground. Food had to be sealed out the way. According to the Hawaii Ant Lab, the amount of fire ants in colonies that my mentor had come across had been there for years, and it was one of the worst cases they have ever seen. Another one would be the invasive crawfish and prawns that flood the rivers, competing and preying on native plants and animals. As for the kalo, they burrow in the corm, cause more holes, more bacteria. Then, of course, you have the wild horses, and then, of course, wild pigs that also do damage. And then, of course, humans. We have our common human and tourist issues as well, too, of people just going down there. And because there's no signs down there, there's no one telling you anything, so it's kind of confusing. So this is a picture right here. On the far left is the Tahitian prawn, or the Macrobranchium lar. So that right there is one thing we captured. This is an example of a homemade trap that we did, that she made and we used, which made with homemade traps to capture the multitude of crawfish and prawns, such as the giant Tahitian prawn, utilizing invasive mullet caught from the streams as bait, a great innovative way of invasive removal. Now, earlier I mentioned when a river jumps, a phrase said by my mentor and her father, Uncle Nale, and I'm sure it's said down in Waipio all the time, but it's a phrase that stuck with me, and it means when a river is suddenly diverted or changes direction due to a tree falling, landslides, or flooding, and it stuck with me so hard because it reminded me of life too. When obstacles fall on our path, we jump. We must adapt to the changes. I have learned so much from my mentor this summer, from her ohana and from PIPES ohana as well. Not only about Waipio Valley or traditional lo'i restoration, but it reminded me what it means to be a Hawaiian, a planter, and a stronger and better person. I learned about time management, whether site access is always an issue as well too, especially when there's no reception, and the importance of being in a community and a community member, not only when in need, but to work together. With all this monotony and growth, I hope to pay it forward, for education is one of our strongest tools and first steps in becoming a better community. Also, to how to learn to sustain ourselves as people of Hawaii, agriculturally, culturally, and educationally. At this time, I'd like to acknowledge and mahalo piha dearly to my mentor, I'imik, my kalani kakakalau, her ohana, Auntie Ku, Uncle Nale, everyone else for having me and teaching me so much, letting me pick their brains for hours and questions. I'd like to mahalo piha to the PIPE staff, ohana, Mina, Sharon, Devin, everyone, including my cohort, for being such amazing inspiration, awesome supporters. UH Hilo for having this program, Kapapa Lo'io Ke'li'ikua Aina, and Waipio Valley for being my classroom this summer. Kamana Girl, she was our mascot, little dog and mascot, at the Lo'i. She was always taking care of us, and I'm truly blessed to have such amazing people in my life and such an amazing opportunity. Oh my God.
Mahalo nui, Jessica, for your awesome presentation. We actually don't have any time for questions, so we're just gonna jump into our next presentation by Sophia Smith.
Hi everybody, my name is Sophia Smith. I am a student at the University of Hawaii at Hilo, and I'm also a PIPES intern this summer. And my project is working on investigating the role of ambrosia beetles in the spread of rapid ohia death under the mentorship of Dr. Helen Sofair with the USGS. So ohia are a critically important endemic tree species. They comprise up to 80% of remaining native Hawaiian forests, and they're one of the first species to start growing on fresh lava flows. They provide important habitat for many other native species, including the honey creepers, lots of native insects, and other native plants. They're also culturally important as well. And I actually grew up myself in an ohia forest, so I'm a really big fan of ohia, and I'm really grateful to be working on a project that helps them out. So, rapid ohia death, which is often abbreviated as ROD, is a disease caused by the fungus Ceratocystis. There are two different species, Ceratocystis leucoohea and Ceratocystis huliohea. So the fungus colonizes the tree's xylem, which is kind of like the tree's veins. It's how they transport water and minerals throughout the tree. And as it colonizes the xylem, the leaves start to turn this distinct reddish-brown color, which you can see in this photo, as well as some of the other stages of ROD. And it eventually loses all its leaves and the tree dies. So ROD was first identified in Puna on the Big Island, and it has unfortunately also spread to the islands of Oahu and Kauai. So we understand how ROD is spreading inter-island. It's likely due to people transporting ohia around the state, and we've managed that by requiring a permit to transport ohia inter-island. But understanding the spread of ROD on a smaller scale is also really important to mitigate the spread, and we're not super sure of how that's happening right now. We know that ungulates like wild boar are a huge risk in the spread of ROD because they wound the trees and leave them vulnerable to infection. We know how to manage them by putting up and maintaining fences to keep them out, and that's shown to be pretty effective. But you can't fence out a beetle that can fly, and we don't know the effect that ambrosia beetles are having on spreading ROD, which is where my project comes in. So ambrosia beetles are a wood-boring beetle that produce frass as they burrow into the tree. Frass is kind of like a sawdust that they expel, and often it sticks to the bark of the tree, which you can see in the bottom photo right there. In a 2020 study, it was found that frass from ambrosia beetles here on the Big Island can contain viable spores of Ceratocystis, the fungus that causes ROD. So they could be spreading ROD in either two ways. They could be producing frass, which contains Ceratocystis, and introducing that into the environment, possibly spreading it in the wind, or they could be carrying spores on their body as they journey from a sick tree with ROD to a possibly healthy tree. So my project looks specifically at two species that use ohia as a host. One of these species is actually native to Hawaii, which I did not expect when beginning this project, and that is Xyloborus similimus, which is the beetle on the top right there, and the alien Xyloborus saxonii is the other species I was focusing on. So the questions I had for this project were figuring out if the beetles were more concentrated in one area than the other, because if they were, that would indicate that something was attracting them to certain areas. And to be able to quantify that question, we asked, do some beetle traps catch more beetles than others, which I'll go more into the traps on the next slide. And then if the beetles are more concentrated in certain areas than others, we wanted to know if they're more concentrated in places where the ROD trees are more concentrated. Because if there was a relationship between them, that would show that, they might be spreading it. And lastly, we wanted to know, since we know that frass can contain viable Ceratocystis spores, we wanted to know how often the frass was containing spores that could create a new growth of Stratocystis. So our field site to investigate these questions was a fenced area of Ola'a Forest Reserve on the Big Island. It's owned by the Hawaii Volcanoes National Park, but it's not inside the publicly accessible part of the park, but it is up in Volcano. So we had 30 beetle traps set up in the arrangement, which you can see on the left. They were set up in a 75 by 75 meter grid, and the image on the right is what the beetle traps actually looked like. So they contained a lure of ethanol because ethanol is similar to the chemicals that trees give off when they're under distress, and those chemicals are how beetles find distressed trees to attack. So the ethanol was at the top of those panels, and it would lure the beetles in. They would hit the panels and then fall into the bottom, which had a cup of water to collect them. And we would take those samples weekly and take them to the lab. So in the lab, the beetles were identified by species and counted by the individual traps, so we had a running tally of how many we were catching. We also received some data from the UH Hilo Spatial Data Analysis and Visualization Lab that included the location of suspected and confirmed rod-positive trees. And with that data, we were actually able to locate those trees in the field. And when we found them, we assessed it for how far along in the stages of rot it was, if there was any noted beetle presence, and if we could find frass, we took 2 samples from trees. I then took the frass that we sampled into the lab, and I carrot baited them, which is essentially just making a frass carrot sandwich. So you put the sample between 2 coin slices of carrots, and you wrap it up, you give it some water, and you leave it in a Ziploc bag for a couple weeks to see if anything grows. So this is a map we made using the SDAV lab data that was generously shared with us. So the locations of our beetle traps are in blue, and then the location of suspected and confirmed rod-positive trees are in red and orange. The red means that it was identified by the SDAV lab to have a high probability of being rod-positive, and the orange means that there's a medium probability of being rod-positive. And of these 23 trees that they identified, the National Park Service actually went in and sampled 11 of them, including both the high and medium probability trees. And all 11 of those tests came back positive. So for the sake of our study, we assumed that all of the trees they had identified were positive for rod. And we predicted, looking at this map, that the traps closer to the rod-positive trees would catch more beetles of our focal species than the other traps. So this map shows the mean number of our focal beetle species caught by a specific trap. The larger the dot, the higher the mean. As you can see, there's definitely variation in the amount of beetles caught by a trap. So we do know that some traps are catching more beetles than others. But the red dots are where the rod trees are located, and just looking at this map, it really doesn't appear to be a lot of correlation between the location of the rod-positive trees and the traps that were catching more beetles. So to examine this relationship a little further, we created this figure, which shows the mean number of focal beetle species plotted against the distance to the nearest rod-positive tree. And as you can see, there's not a strong relationship between them. We also did a linear regression model on this, and we found no significant relationship between the mean count and the distance to the nearest rod-positive tree. So to look at how often the frass we were collecting contained viable Stratocystis spores, I left my carrot sandwiches alone for several weeks to grow whatever fungus or bacterial they could. And I had 30 samples in total, so 13 from confirmed positive trees, 13 from suspected positive trees, and then we had 4 samples from field-identified suspect trees, which we found during the course of the study that when we were going from tree to tree, we would sometimes find other trees that appeared to be symptomatic of rod that had not been identified by this SDAV lab, but we sampled some of them anyways just to see. So when we had the samples sitting for a couple weeks, I would set aside any of the carrots with black-looking growth, because that's what Stratocystis looks like, and then we would look at it under the microscope. And this is what rod looks like under the microscope. You can see the black growth, and you can see that there's these like hair-like projections that are easy to identify, so it was easy to identify the rod. And then I had my colleague also help me do some microscopic identification, and she also did genetic testing on the samples to confirm that it was indeed Stratocystis. So what do these results mean, and why is there no relationship between these variables? So there's several reasons that there could be no relationship between the rate of beetles caught and the distance to the nearest rod tree. The biggest issue I found was that there appeared to be many more rod symptomatic trees in our study site than had been identified by the lab. And since that was unaccounted for, we didn't have that in our metrics. This could have been because the aerial survey data we used was from last year, so there could have been more outbreaks since then. And it also might be because SDAB is more conservative in their counting of the rod trees, so they would rather underestimate than overestimate the amount of trees. An updated aerial survey or even a comprehensive ground survey of all the ohia trees in our study site would probably give us a better picture of the relationship between the rod and the beetles. And then also, beetles aren't only interested in the trees affected by rod, they're interested in any tree that's undergoing distress. So a tree dying of natural causes could also attract beetles, which we did not account for. And we also just don't know how far the beetles can fly from one tree to another, so they could be less localized just because they have high mobility and they don't just stick around their local tree area. Or it just might be that these beetles aren't very closely related to rod trees because they might not be a huge distributor of rod. We definitely need more data to make that clear conclusion, but looking at the results of the frass sampling, as you saw, like even the trees that were confirmed positive with rod, not every sample contained viable spores, not even most of them. So it looks like right now that they're not a huge risk, especially compared to other risks such as ungulates, wild boars. Sorry. It won't let me go to my next slide. It won't let me go to the next slide. Is there something I can do? I'm pressing it. It's not going. It's just highlighting stuff. Oh, sorry. It just froze. How do I present it?
View and slideshow at the break.
Okay. Sorry about that technical difficulty, but I wanted to thank my mentor, Helen Sofair, for all her guidance on this project, as well as all the other team members of the Beetle Project, Bob Peck, Ellen Dunkel, and Jordan Zarders for all their help at this time. I'd also like to thank Tim O'Sullivan of the SDAB Lab for sharing the data with me. It was super valuable to my project, and I'd like to thank the entire PIPES Ohana for all their support this summer, and I'd like to thank everybody for listening. Do I have time for questions? Yeah. Okay.
Did you notice an specific on the tree where the beetles were attacking? Would you say it would be like lower around the trunk? Would it be higher?
That's a really good question because one of the limits of our study was that we could only sample where we could see on the tree. We weren't cutting the trees down, so we couldn't see very far up, and I'm not a very tall person, so I was pretty limited in my observation. So we could only see from about like maybe a couple feet above our head to the ground, and that's where we found the most beetle presence, just because that's where we could observe it. Thanks, Lehua.
Wow. What did you learn about working for the feds within the world of conservation?
Well, it was a really good experience. Before this summer, I was really nervous to kind of take the leap into a professional field because I thought I didn't have what it takes to be a real scientist. But working with this, I was really grateful because I felt like I was working with really just cool and knowledgeable people, and it gave me a view into what like the day-to-day life would be like as a scientist, and it made me realize that this actually is something I would be interested in pursuing as a career, and it made me feel like I could actually do it.
Great, thank you. All right, moving on. Thanks so much, Sophie.
All right. For our next presenter duo, we have Minnie and Kaehukai, who will be talking about their research done in Kalapana this summer. Just a little backstory, the two of them both worked in the same area of Kalapana, but had two different kind of areas of focus, if you will. I don't want to give too much information away, but they'll be giving the presentation together and talking about how their work connects and how it relates to Kalapana. So without further ado, I'll give it to Minnie and Kaehukai.
Aloha everyone, my name is Ka'i'ini Pu'uvai Keli Ho'omaluhos, but most of you guys know me as Minnie, and this is my partner, Kaehukai Goin, and our presentation title is Malama Ika'aina Momona o Kalapana, which Kaehukai will elaborate more on.
Aloha mai kākou. So our title means to take care of the fertile land of Kalapana. So we created this title as a representation not only as Kanaka, but genealogically and culturally speaking, looking to the generational resources of why it's important, yeah, and the mana'o of Aina Momona, right? The fat, the fertile, the fruitful land, right? This is the Hawaiian worldview in which we did our research, mixing modern techniques with our ikikupuna as we studied the coastal ecosystems of Kalapana.
So we'd like to start by giving a big mahalo to our mentor, Gina McGuire, and everyone who has helped with this project this summer, including the PIPES Ohana, Sharon Ziegler-Chung, Mina Viritua, Devin Aguiar, Noelani Puniwai, Lisa Kanale, Rebecca Ostertag, Dr. Carla McDermott, Dr. John Price, and the Kalapana community, including the Keli Ho'omalu and the Hawai'i families.
So our kulana, or positionality for the project, my kulana from this internship really helped to define my kuleana as a Kanaka in this modern day society. I originally come from Oahu, and I have a background in Hawaiian Studies, Malama Aina, along with Marine Option Program from the University of Hawaii at Manoa. Growing up in this concrete jungle, it created a disconnect within myself from Aina, Kupuna, and just my Hawaiian identity. So through my education pathway, it helped to reconnect this pilina between Aina and helped me to learn why we need to malama these resources for the next generation so that these resources will always be able to take care of them.
My kulana, being raised in Kaimu Kalapana, a rural community on the eastern coast of Hawaii Island, I have experienced firsthand how important family and my lahui are. Through my upbringing, I was instilled with the knowledge and passion for supporting Hawaiian cultural practices, and as a Hawaiian wahine, I know that modern life is neither kind nor soft. In fact, the political, economic, and cultural reality for most Hawaiians is difficult. So I would like to help care for Aina and our community, because central to the Hawaiian culture is a strong stewardship ethic of people and place. Hawaii's unique and precious natural ecosystems form the very foundation of this culture, as well as Hawaii's economic stability. So with that being said, I hope to continue spending my time preserving and restoring our native ecosystems for future generations to come. As you can see here, our study site is bordered in yellow up there, and it's roughly 5 to 6 miles long. And we identify it as Kalapana Nui on the Big Island of Hawaii in the Puna area, and it stretches from the boundary of Kehena Ahupua'a in the east to the boundary of the Hawaii Volcanoes National Park in the west, and consists of Kaimu, Kalapana, Kikala, Ke'okea, and Kapa'ahu Ahupua'a. This rocky coastline consists of marine habitats thriving on cliff faces, boulder beaches, and rocky tide pools, as you can see in the top left-hand corner picture up there, alongside native coastal shrubland and remnant agroforest coastal vegetation communities, as you can see in the other three pictures to the right. Due to the frequent volcanic activity of Kilauea throughout history, the land area of Kalapana consists mostly of these shallow organic soils. And Hawaii Island, being the youngest of the Hawaiian archipelago, has shallow organic soils that have formed over young lava flows. For example, the Kalapana coastal communities, which are built on Pahoehoe lava flows from 1986 to 2018, where there is still little to no soil, which makes this land hard for farming. However, this Aina makes up for this in its abundance of coastal resources.
So the history of Kalapana shows the importance of the people and its coastal resources. But why are coastal ecosystems important? Anybody want to spit anything out? Lenin, can we get someone not on why coastal ecosystems are important?
The question was, why are coastal ecosystems important?
Yes.
Originally, Native Hawaiians settled on the coastlines before moving inland as the population grew.
Yeah, exactly. All right, so for Kanaka, Hawaii, we depended on the health of our natural resources, in particular the lowland and the coastal resources, which is why there were so many community settlements throughout the Pai'aina as coastal communities. But looking worldwide, coastal ecosystems are one of the most diverse and productive areas that rival even the productivity of rainforests for raw materials and goods used by humans. For the communities of Kalapana, it provided the aulapa'au, coastal invertebrates like opihi and lobster, fish like uhu and paku'ikui, helped with the gathering of salt and limu, as well as transportation with many canoe landings along the coast. But looking further into our study of the importance of coastal ecosystems, many studied the coastal vegetation of communities, and I studied limu species, density, population along the Kalapana cliffs, and in the accessible tide pools.
So our goal has been to take the time to document this coastline's vegetation and limu communities in the hope to preserve and restore our native ecosystems while fulfilling 4 native Hawaiian values. These values being 1, to hanai, to foster and care for the po'e kanaka and ka po'e holoholona, so the people and the animal people of Kalapana. 2, ma'oli no, placing endemic and native species as priority. 3, hi'iaka, encouraging new growth grounded in wellness and in the knowledge that people are important factors in rebirth. And 4, pono, practicing being conscious and considerate in all that is done.
So for our methods, we did limu observations, vegetation surveys using the brown blanket method, which provides habitat structure and species abundance. And as well, we did ethnohistoric cataloging. So we first started our project by reading and researching mo'olelo from our kupuna, like Auntie Emma Kauhi, who was from the Kalapana area. And the reason why we did this is because as Indigenous and Pacific Island tradition, the role of mo'olelo and storytelling is crucial information for us to use. Sorry. And from these stories, it helps to guide our decision making, especially in the Malama Aina realm. We also dove into new Pepa articles that mentioned limu and la'au from the Kalapana area. And from there, the stuff that we catalog, when we went into the field, we concentrated on the populations of one species. So limu kala was the most prevalent, and then there was other mixed vegetation communities that many went into. So this is the brown blanket method, and we're starting off our first transect. So using this line, it goes 30 feet in, and then we'll just study what the vegetation is within the 1 square. This is one of the many New Pepper articles that we looked up. This one mentioned Limu lipoa as a prominent limu species back then. And then we also were gathering limu in the tide pools of an undisclosed area because it has plenty resources.
In addition to documenting the historic vegetative communities, a current inventory of Kapo'ela'au was completed for the immediate cliff vegetation community. And we also used the ArcGIS, so it's a geographic system, to create a map of the current research where proposed agroforestry restoration may be suitable, as you can see on the left, and then our site area highlighted in pink. Potential restoration community composition was also visualized using the agroforestry design tool. As you can see on the bottom, that little picture of there, which is based off of a hybrid approach of methods and tools. These include ethnohistoric reconstruction, functional trait analysis, cultural significance, and modern agroforestry production design guides. And together, these methods allowed us to constellate an agroforest that is inherently biocultural and informed by multiple perspectives. And as you guys can see, the table to the right shows some of the several species that were recorded historically, along with their functional traits and where they're found. We captured the ethnohistoric accounts of around 83 Kapo'ela'au species, all of which were referenced by Kupuna and authors regarding Kapo'ela'au of the wider Kalapana district, including a variety of elevations and plant types.
In my findings for this internship, we looked at how limu kala distribution relates to the coastal age on this lava-impacted coastline. In the first map, we got data from the Arizona State University geologic map, and this was of Hawaii for the substrate age. The purpose being that the age of the substrate determines certain species habitat and limu growth. When accessible in the tide pools, limu kala presence and absence was recorded with a GPS device along the boulder cliff communities of Kikala, Keokea, Kaimu, and Kalapana. Limu kala presence and also their absence points were documented every 100 meters. Our duration for observing limu kala was between June 7th and July 30th, 2021. Representing a little snapshot of limu kala within its seasonal and long-term life cycles. The second map is on limu kala data we collected, which, as you can see, shows no abundance on the younger coastline due to the 1986 and 1992 lava flows. In particular, the youngest lava flows are area in the west of the study area. When we observed limu kala, it was more abundant in blanket patches with higher concentrations to the east of the study in Kaimu. So like right here is where it starts and then goes up. And the reason for this is like this side was more accessible, so we could determine like positively the limu kala. Whereas, not saying that there wasn't limu kala over here, but there's only certain areas we could actually 100% identify it was limu kala. Just also in the difference of like, this coastline was like 70 feet cliffs, 60 feet cliffs, whereas this was more easy and attainable as well. That was another factor in the limu kala research.
Moving forward, we hope that this can guide future restoration work informed by both Ike Kupuna and modern ecological methods, as well as the preservation of these ancestral resources for future generations. Thank you all for your undivided attention. Any questions?
Okay, so it's a question for both of you. What would you say was your favorite thing that you learned or did working in Kalapana?
Mine was definitely doing the field work and getting to work with our mentor and him in the field. And just like because I live down in Kaimu, so just being able to like walk along the coast and know what's there, the plants and the animals, the limu, it's always fun to learn more about.
For my favorite part, it was just being able to be outdoors and in a place that isn't heavily populated, and you pretty much get to see like life restoring itself, you know. It's an amazing feeling, and just the coastline itself, like it is Momona in coastal resources, but like it humbles you because at any time, if you get hurt, you really have no help out there, you know. So, yeah.
In your observation,
Yeah. The question was if we saw people coming to gather limu kala from when we was doing our study. So when we were doing our study, like I said, like there was almost nobody out there. We saw maybe like a couple fishermen this summer, which was why it was also a super amazing experience, you know, just being out there with no human interaction except us. But Minnie, I don't know, have you seen people?
No. Yeah, like he said, when we're doing our study on the coastline, I would say especially like just because it's mostly cliff along this coastline, like he said, tall cliffs to short cliffs, but it's like getting into the water or just like easily accessing the limu would be like harder. Yeah, unless it was like tide pools and stuff, but still the high surf or wave action makes it like harder to collect it, so. Any other questions?
I know that you just said that it's kind of dangerous to gather over there, but I was wondering if in other places along the coastline you were planning on sharing this information to the community in order for them to be able to access those resources, if they didn't already know that they were there.
So for like our project, we did make like a bunch of maps and like lists of like the different limus and the plants that we found, and I'm sure that would be made available to everyone and the Kalapana community, but right now I'm not too sure if it's out there right now, but yeah.
Another question.
One more question.
Sorry, you guys don't get off that easy.
Mahalo, Sharon.
Anytime. So this project is really cool in its engagement of so many different forms of knowledge. And so what do you see, what are your thoughts after having worked out there and you're doing data collection, you're producing maps, you're using Kilo, you are interviewing people, you're talking, you're looking at Nupepa. What's your feeling on this? Is this a way that we could apply, an approach that we could apply to other communities to help them understand and better manage their resources?
So pretty much the research that we were doing is like gathering our arsenal so that we have all this information, and from my perspective, pretty much to protect the resources from like development. You know, nowadays it's getting harder and harder to even get limukohu, like, or even kala. Like, this place is, it's hard to describe it in words. Like, we can say that it's just really hard to access, but you know, our kupuna had to do it. So, you know, if we had to, we can definitely do it. But just the fact that all of these resources are here, that's important enough, I feel, to protect it. Whereas, you know, Oahu, where I'm from, growing up in the concrete jungle, like, where are you gonna find kala? Where are you gonna find kohu? Like, you know, so pretty much just growing the arsenal against development. Any more? Oh, most definitely, yeah. Yeah, can you repeat the question? Can you rephrase the question for me?
Using this methodology, so where you are combining these different ways of collecting information, so many different things. Do you feel like this is a methodology that could be applied to other communities?
Yeah, most definitely. You know, the whole point of this program, right, is to pretty much bridge our culture with, you know, Malama Aina, and how do you explain it? I don't know, for Hawaiian studies, it's kind of like common sense already. I don't mean to offend anybody by that, but yeah, it's like a cultural impact assessment along with everything, nature, like, yeah, I don't know how to like further on that.
Just like, especially using like the mo'olelo or the ike kupuna, like we see, well, for my side of the project more, so we see like what vegetative or plants were there before, you know, native plants, and like because there's a huge problem with invasive species now, you know, so just like referring back to what plants were there before can definitely help, like when you're trying to restore that place, like what kind of plants you should replant, or, you know, look more into planting, because that's like the plants that grow there or where they're from, yeah. But yeah, I definitely think it could be used for other sites as well.
Perfect, that's the answer I was looking for. Huh. Wonderful.
Thanks, Sharon.
Done. Lunch break.
Yo, lunch.
Aloha, everybody. We are coming back from our lunch break now. Mahalo for our virtual attendees. Thank you for joining us again. We're going to be picking up our talks. We have 3 talks scheduled before we'll be taking a 15-minute break. The first person that will be presenting is Taylor Ui Barangan, who got to work with the USGS Pacific Islands Ecosystem Research Center this summer. And she'll be sharing some of the work that she's done to kind of help promote the health of our native birds, working with the mosquitoes. But without further ado, I'll give it to Ui to present.
Aloha mai kako. My name is Uilani Barangan. So this summer, I was so fortunate enough to be working with USGS on a project for avian malaria, comparing trap types for capturing avian malaria positive mosquitoes on Hawai'i Island. So we are so lucky that we live on an island with such beautiful creatures. The Hawaiian archipelago is home to 18 beautiful species of honey creepers, many of which are single island endemics. Traditionally, their feathers were used to decorate the ali'i, or the chiefs of the land, to signify status. In addition, many of these birds were thought of and are still thought of as guardian spirits to the forest. Originally, there were 56 different species of honey creepers, but now there are only a mere 18 left due to rats, ungulates, cats, lots of invasive species, but the biggest one is mosquitoes. So if we want to keep our ecosystems and Hawaiian culture alive, we really need to start focusing on the health of our honey creepers by studying mosquitoes. Mosquitoes. They really suck. They are vectors for diseases such as malaria, Zika virus, and dengue fever in humans. But they're also big transmitters of avian malaria and avian pox in honey creepers. So because honey creepers evolved in an isolated archipelago, they lost the resistance to avian malaria. They're completely helpless. Additionally, many of our newly introduced species, such as Japanese white-eyes and zebra doves, they're able to harbor avian malaria as well, which furthers the spread to our honey creepers. So what causes avian malaria? Well, there is a parasite that's called Plasmodium relictum, and basically, here's the diagram up here. A mosquito will bite a bird, and that bird is then infected. And when another mosquito comes along to feed on the bird, that translates into the next mosquito, and it goes through their salivary glands and affects the whole body. So honestly, it's not really just the mosquitoes that are the problem. Like, there's a deeper issue, which is the Plasmodium. So I need to tell you this right now before I get into anything any deeper. So there's a big difference between the two infection sites of a mosquito. It can get infected in the thorax and the abdomen. Now, if it's infected in the abdomen, that means that the Plasmodium is not quite active, so that mosquito can't necessarily infect another bird. The eggs in the mosquito might become infected, and that might be an issue, but if it has an infection in the thorax, that's where the salivary glands are. So when the mosquito bites the bird, that's how the disease transmits. Now here's a graph that translates to elevation and the abundance of vectors, parasites, and native birds. Now, you'll see that in lower elevation, that is where vectors are most common. So if you live in Puna area and Hilo, those are lower elevational areas where you're more likely to see mosquitoes. And as you can see from the graph, from roughly 500 to 1,500 meters, malarial parasite populations peak and vector populations decrease, and native bird populations are at their lowest. Now, vectors appear to be most common in the 100 to 400 meter range and steadily decrease until the 16-meter elevations. Now, native birds are starting to reside in higher elevations because they're trying to get away from all of the mosquitoes and the malarial vectors. But eventually, they're not going to have anywhere to go. So due to climate change, all the temperatures in which many of the mosquitoes and malarial parasites can survive, they're going to start moving upward because things are becoming warmer and they're going to be able to survive. And you can already see there's an overlap with peaks. So where malarial parasites peak, but the vectors start to decrease, those are the very resistant malarial parasites that hang out in that area, which is incredibly unfortunate. And I should also mention that parasite reproduction with Plasmodium relictum does fail at an elevation of 1,800 meters, but it's most rapid at 300 meters. But all of that is expected to change in the near future. Now, are any of these birds completely helpless, as I've mentioned? Well, remember how I said because honey creepers evolved in an isolated archipelago, they lost the resistance to avian malaria. Well, there are some species that have a very, very, very slight resistance to avian malaria. Luckily, our amakihi has a resistance estimated to be roughly around 20% for the projected rate of declination in higher elevations, which is great news. And roughly 30% of cases, primarily with apapane and our amakihi, birds with low intensity chronic infections are able to become relatively resistant to reinfection. So they can't maybe get infected twice. But unfortunately, our EEV bird, which is what most people think of when they see a honey creeper, has the highest mortality rate of the Hawaiian forest birds with a severe 10% survival chance. Now, we have a lot of current solutions for fixing this, but unfortunately, a lot of those solutions are going to take time. Some of our solutions include vaccinating the honey creepers against avian malaria, genetically editing them so that they're resistant. We also have another solution, which is genetically editing the mosquitoes so they're unable to reproduce. And of course, we have our non-environmental friendly solution of just pesticides. And as I said, a lot of these solutions are quite difficult. But in the next 2 years, Hawaii is looking to use the IIT technique, which is the incompatible insect technique, which is basically trans-infecting mosquitoes with naturally occurring bacterial strains of Wolbachia that act as a birth control for mosquitoes by making them relatively infertile. So this is an image of where my samples were collected. This was taken from the Waiakea Forest Reserve. And up there's kind of a star in the upper right corner, and that's where this whole radial quadrant is. So the hypothesis of my study was we have 2 different trap types, and is there a difference between them in catching mosquitoes? Are they going to have different proportions of infections? Well, for my project, I collected 48 samples, and from these samples that were taken from here, you can see kind of the little boxes and the triangles. That is from where the traps were spaced. They were spaced 30 feet apart, located at around 900 meters in elevation, and laid out radially, not elevationally. And they were put in a mark and recapture and release study, and we took a lot of these samples as our own, and these were taken from Dennis LaPointe and Carter Atkinson. So this site was chosen primarily for convenience, but also because this site is awesome for studying mosquitoes and native birds, since it's in somewhat of a low elevation, but it has a big gradient in where you can take samples from. So these samples were dissected, and then DNA was extracted from them and analyzed via qPCR. So these are 2 different types of traps. Now, remember how I said that abdomens and thoraxes are very different? Well, on our right, you can, I think that's your right, there's our gravid trap, and that is primarily for catching mosquitoes who have eggs. So they've already had a blood meal, they've already fed off of a bird, and they're likely to be infected with Plasmodium relictum. On the left is our BGS trap, and that is for capturing mosquitoes who are looking for a blood meal. Now, what's interesting about the BGS trap is that because there are mosquitoes looking for a blood meal, some of them might not have ever had one. They might just be brand new, they're looking for blood, and they're ready, but they might not have become infected yet. So due to the difference in life stages, it was hypothesized that gravid traps are more likely to catch mosquitoes infected with avian malaria because they're looking for blood and have already had it. So I had a lot of steps in this process. So I had 48 of the samples, but they were dissected into 3 different parts. So the heads we didn't use, but we used the abdomen and thorax, so a total of 98 samples. And basically from there, I was able to cut them up and then extract their DNA, which was such a cool process to see how that was done. And we used a Qiagen DNeasy kit. And from there, I was able to perform qPCR, which is a technique that's currently being used a lot. Any guesses in what it might be used in for identifying? COVID. So PCR is a big test for COVID. And basically what it does is it amplifies what we're looking for. So here we were looking for the presence of Plasmodium relictum in these mosquitoes. So once it identifies it, it cycles it several times. and from there it amplifies it. So we can visually see if this mosquito has DNA of the Plasmodium relictum. And each sample was tested 3 times just so we could be sure about these. And as for our results, the proportion of mosquitoes that tested positive for Plasmodium relictum approached significance using a chi-square test, which showed a p-value of 0.051. So we were very, very close for approaching significance. So we can't necessarily say that one trap was more effective than another, even though we've like hypothesized it. But again, this is preliminary data, so we'll have to look into it a little bit further. Now, what can you do about avian malaria? Because I'm sure you guys are all concerned about honey creepers now. So research has indicated that avian malaria is increasing in upper elevation areas, such as Hakalau, but some research contradicts that. But either way, it's likely that the proportion of avian malaria will continue to grow, as well as the probability of infection of Plasmodium relictum due to the preferred gestation conditions changing with climate change. So we have a lot of hope for the incompatible insect technique to control mosquitoes, but what you can personally do is reduce the amount of standing water you see around. Humans have a huge influence on standing water, whether it be from old tires or pots, especially ceramic pots in a garden, because they can hold a lot. And lack of breeding areas will translate to the reduced transmission of avian malaria, which can possibly help our birds out as much as possible. So I'd really like to acknowledge my mentor, Renee Bellinger and Stephanie Mladnick. They were just so amazing, and I've loved every step of the journey. I'd also like to acknowledge Carter Atkinson and Dennis LaPointe for graciously letting me have their samples to cut up and use for my little experiment. And I'd also like to thank USGS, NSF, and of course the PIPES Ohana for their support. Mahalo, you guys, and please let me know if you have any questions.
Is there any trend in which honey creepers have more resistance to malaria or not?
If they have more resistance?
Yeah, because you said there was a difference in like the amakihi versus the EEV and how likely it is they'll suffer from it or get severe effects.
That's a good question. I would personally like to explore just like the elevation between those. I think that would be a really good place to start. But as far as I know, I don't know if there's really quite a difference between those two birds and why the EEV is more susceptible to infection. But that's a great question.
You had said that you went through a dissection and DNA extraction process. Did you have to do that a lot? Like, was there multiple times?
Oh my gosh, that took the most of my time. So I had to basically cut open each mosquito 3 times and extract DNA from both the thorax and abdomen. So totaling, like, it probably took more than like 10 hours to do all of that because it was a lot of samples, and like I would have to do them in like maybe sets of 10 just because all the materials we were using were really time sensitive. So if I'm doing batches of 10, you know, it takes a really long time. But yeah, good question as well.
Any last questions before we transition to the next presenter? Alrighty. Next presenter up, Isabel Schwartz, will be traveling to Keoho on the Big Island in Ka'u, and she'll be talking about her project working with the Three Mountain Alliance.
Aloha everyone, my name is Isabel Schwartz, and this summer I worked with Three Mountain Alliance in Volcano, in the National Park, and in Keoho. And we decided to do a project on seed treatments that could possibly deter rat predation. So this project focuses on Iliahi, or Santalum paniculatum, and this is a native plant to Hawaii. It is hemiparasitic, which means it needs to be grown near a host plant for certain nutrients. And it was also over-harvested in the late 18th century. Within the heartwood of the trees is a fragrant oil, which was a very valuable trade item. And this process was very ruthless on the people of Hawaii. The Maka'ai Nana, or the common people, were the ones that would have to harvest these trees, and they would carry logs on their back and walk great distances that they were referred to as kualeho, or callous backs. And Iliahi also has a very long dormancy and germination period. The seeds can be dormant for up to 2 years, which means they aren't germinating, but they're still viable seeds. And they can also take over 4 years to germinate. And in the future, Three Mountain Alliance would like to do a direct seeding method. And this basically means you could collect the seeds and then clean them and place them back out into the wild, instead of having to collect seeds, clean them, grow them in a greenhouse until they're ready to be outplanted. So a direct seeding method could save time and money. And Iliahi would be an ideal plant because of the long dormancy and germination, which makes it difficult to grow in a greenhouse. So prior to human contact, there were no native rats or mice in Hawaii. And rats damage native plant and bird species. They love to eat Iliahi seeds. So on my first day of work, my mentor Emily and I cleaned about 6 pounds of fruit, which resulted in about 2 pounds of seeds. And to clean the seeds, we used our fingers to remove the pulp, which you can see on the paper plate on the bottom right corner. And the clean seeds are the ones in the blue bowl. And this took about my entire first day of work, and at the time, I didn't know that we would end up using these seeds for my project. So for my setup, we had 4 different locations, and at each location, there was one enclosure. And there's a diagram of the enclosures on the right, as well as an actual photo. And within each enclosure, there was a small door to prevent larger animals from entering, and then also to allow rats or mice to enter. There was also 2 yogurt cups in each corner, one with control or untreated seeds, and one with treated seeds. We split the experiment up into 2 treatment weeks. The first week tested peppermint oil, so we sprayed seeds with essential oil with peppermint oil. And the second week, we tested squirrel sauce, which is soybean oil and chili pepper extract. And we also strapped Moultrie A40 Pro game cameras to nearby trees in order to take pictures of our enclosures. And each morning, we would check all 4 enclosures to see if any seeds were missing. If seeds were missing, we would add seeds. So every morning, there were 10 seeds in each yogurt cup. And every other day, we would bring a laptop to check or download our pictures. So during the first treatment week, there were no seeds missing or confirmed rat visitation. This means that all the seeds we put out on day 1 were still there on day 5 at all 4 locations. The second treatment week is where things get more interesting. On the second night, there was a confirmed rat at location 1. And every night after this night, there was continuous rat visitation. So the table you're seeing, this is for treatment week 2, the squirrel sauce and the control. And this is only for location 1, because this was the only one with a rat visit. So on day 1, all the 10 seeds were there, and that night was the first night a rat came. And they only ate 4 squirrel sauce seeds, so we had some hope that the squirrel sauce was working. However, as you can see, by the end of the week, they ate both the control seeds and the squirrel sauce seeds. So because there was repetitive rodent visitation at location 1, we decided to put out 2 enclosures at this location, We put one with squirrel sauce and control seeds and one with peppermint and control seeds for 3 days and 2 nights. This information would tell us if the rats simply hadn't found the seeds that first week or if the peppermint treatment was effective. And unfortunately, all of the seeds were eaten after we checked 4 days. And we also have photos from the game cameras confirming rat visitation that I'll share with you guys now. I thought it would be fun if you guys could try and spot the rat yourselves. This one is pretty obvious because the rat is staring right into the camera in the center. And this was taken the first night that we had rat visitation. This one's a little bit harder. You probably noticed this rat first. He's in the peppermint cage. And then this rat is also pretty obvious. He's on the side with the squirrel sauce. But it was really interesting because we were able to capture 3 rats in one photo. So some of the takeaways are rodents are a prominent threat to Iliahi. Our data shows that they're eating the seeds. We lost 94 seeds within 2 weeks. So I can only imagine if you were waiting 2 years for these seeds to germinate, how many seeds you would lose. And we also learned that seed treatments could work as a possible short-term solution. However, once again, in the case of Iliahi, it just takes too long for the seeds to germinate. So it's probably not the most effective, but it could possibly work on other native plants. And then the next step would probably be to test how these seeds will germinate after they were treated, if the treatments had any effect on their germination. And in conclusion, I think that rat eradication or control would possibly be more effective in helping restore these native plants. And I would like to give a thank you to Three Mountain Alliance staff, my mentors Emily and Callie, the PIPES staff, my fellow interns, and my family. I hope you guys are watching. Are there any questions?
Awesome job. In the future, is there anything you'd recommend covering the seeds with aside from like peppermint oil and the squirrel sauce?
Yeah, so I think that I still have hope for peppermint just because the first week they didn't come, so I feel like I would like to see if the peppermint, if rats don't like the scent, maybe in a lab setting you could see that better. And then also trying something maybe more spicy. I'm not sure how spicy that squirrel sauce was, but regardless, they ate the seeds, so I don't know how effective the treatments will be.
I was just curious if your treatments would affect Iliahi germination in any way.
Yeah, so we don't know that yet just because of the span we had for this project, so that would be kind of the next step. We're hoping they don't, but also it would take a long time to know just because they take a while to germinate.
I just wanted to say I don't have a question. You have quite the family following. Izzy has filled up the chat, and I have to say that UI gave us about 15 people watching for a while too. So, anyways, I said that was my good question, which isn't a question. So, anyway, I'm going to pass it on over to Lehua.
Why didn't? I'm curious with the germination of the Iliahi seeds, do you folks do anything to try to speed up the process? Maybe like heat them up, score them, throw them in the freezer to get the germination going?
Yeah, so they do propagate the seeds and they do treatments depending on the seeds. While I was there, I don't remember sowing any Iliahi seeds, so I'm not sure about those treatments, but it could be possible that they do that.
Do you have any idea as to why they were eating the iliahi seeds, or why they were particularly more fond of those seeds?
I'm not sure why iliahi specifically, but I think that seeds in general are a food source for rats. And I'm not sure why iliahi are super prone to rodent predation, but it is definitely an issue with iliahi specifically.
Thank you.
All right. So now we do have a question. So what did you learn about the role of these collaborative organizations like Three Mountain Alliance? What did you learn about their role within caring for our Aina here in general and all of that? What did you learn that?
It's really cool what they do, and I've been to some of their restoration sites in Keoho, and I remember my first day thinking, this is so beautiful. I would have never seen this place, never known this place. And the fact that these organizations are essentially planting native forests, to me, they seem almost like superheroes. Like, it's a really big task, and they're just going out there and doing it. And I think it's cool to know that with the groups and the collaboration, you can get a lot more done as just me going out there trying to plant a native forest.
But without further ado, I'll give it away to Kylie.
Aloha, my name is Kaylee Alcasiba. This summer through PIPES, I interned at the USDA office in Hilo under Dr. Stephen Hess and Carmen Intaki. I will be presenting on the distribution, characteristics, and behavior of the Polynesian rat, the diseases it carries to humans, and its cultural aspects in the Pacific. The Polynesian rat, Rattus exulans, also known as the Pacific rat, originates in Southeast Asia and has since spread to numerous regions in the Pacific. It has been introduced to the East Indies, Philippines, New Guinea, and Pacific islands such as New Zealand, Easter Island, and Hawaii. Through paleoecological evidence and ethnographic accounts, it is suggested that the Polynesian rat was introduced to the Pacific Islands by the first Polynesian settlers. The Polynesian rat, known as Iole in Olelo, Hawaii, is recorded on every main Hawaiian island and was eradicated on Kura Atoll and recently Lehua Island. Iole occupy a variety of habitats. Characteristically, they inhabit lowland forest, agricultural fields, wooded and grassy gulches, and waste areas. However, Iole have also been found at higher elevations, such as on Mauna Loa at about 5,000 feet, and on the rim of Maui's Haleakala crater among nesting petrels at around 10,000 feet. The Polynesian rat is the smallest of the Rattus genus, averaging about 150 millimeters in body length and 40 to 100 grams in weight at maturity. Our exulan's coat is a brownish-reddish color on top and a light gray or white underneath. Slender and agile, with small feet and clawed toes, they are adept runners and climbers, but poor swimmers. The Polynesian rat has a lifespan of about 1 year in which their teeth continuously grow. They must use their incisors frequently to maintain a healthy functioning length. Like many other rodents, the Polynesian rat is primarily nocturnal. However, in higher densities, like on some tropical islands, they appear to be equally diurnal and nocturnal. Polynesian rats are known to be opportunistic omnivores, meaning they will eat available plants and animals such as seabirds, lizards, insects, and native plant species that have not evolved with mammalian predators. They are considered an agricultural pest due to their extensive damage on a variety of crops such as sugarcane, coconut, cacao, pineapple, rice, macadamia nuts, and root crops. Due to the shift of high sugarcane production to a more diversified agriculture in Hawaii, rat damage is now significant to seed crops such as soybean and corn, tropical fruits, and native plants. They are also believed to have played an important role in the major reduction of native palm forests that made up most of the lowland vegetation. In addition to being considered an agricultural pest, the Polynesian rat can either directly or indirectly infect humans via numerous pathogens. Diseases such as the bubonic plague, leptospirosis, toxoplasmosis, and rat lungworm disease can be carried by R. exulans, and if transmitted to humans can be life-threatening. Humans can contract such diseases through rodent urine and excrements, as well as through vectors that transfer infection from rodents to humans. Although the black rat, Rattus rattus, is considered the main host of the flea that carries the bacteria of the bubonic plague, other rodents such as the Polynesian rat and mammals in general can be hosts as well. Referred to as one of the worst pandemics faced by humankind, the bubonic plague has been estimated to have killed over 200 million throughout human history, with cases still being reported worldwide. The plague reached the Hawaiian Islands in the early 1900s through merchant ships. Efforts to combat the spread of the plague included quarantines as well as setting fires to areas identified as needing to be sanitized to control contamination. These fires quickly metastasized out of control in many areas, and in one case destroyed about 40 acres within the Chinatown district on Oahu. Almost 400 individuals were killed by the plague in Hawaii, with the last reported case of infection being in 1949. In this picture is one of the fires on Oahu. Leptospirosis, caused by the spirochete Leptospira intergons, affects around 1 million people worldwide annually, with about 60,000 reported deaths per year. Humans can contract the disease through water or soil that has been contaminated by other infected mammalian hosts, such as R. exulans. Symptoms of leptospirosis include fever, headache, and myalgia. Prior to the 1995 removal of leptospirosis from the U.S. list of nationally reportable infectious diseases, Hawaii had the highest annual incidence rates. This disease is still prevalent in Hawaii, with around 350 cases reported between 1999 and 2008. Toxoplasmosis, caused by the protozoan Toxoplasma gondii, is an infection that occurs worldwide, where rodents are the main reservoir. Cats are the definitive hosts that complete the life cycle of T. gondii by passing oocytes through their feces. Infected pregnant women may experience spontaneous abortions or may notice developmental delays in surviving infected children. Native animals such as nene, alala, and the Hawaiian monk seal are also infected, in which some cases are even fatal. This is the life cycle of the parasite. Rat lungworm disease, angiostrongyliosis, is the leading cause of human eosinophilic meningitis. Rodents are the definitive host of this disease and pass first-stage larvae in their feces, which are then ingested by intermediate hosts such as slugs or snails. Within the intermediate host, the larvae reach the infective third stage. Humans can become infected after ingesting the third-stage larva, or the infected gastropod, or consuming contaminated produce. By consuming contaminated produce, the larvae then travel to the brain. Since 2004, cases of rat lungworm disease in Hawaii have resulted in permanent disability, coma, and even death. Many islands throughout the Pacific, such as Kosrae, New Zealand, Vanuatu, and Hawaii, mention the Polynesian rat in myths and legends, mostly concerning their appearance, behavior, and distribution. For example, the origin of the rat clan in Kosrae involves the rat being a beloved helper of God in heaven. God goes on a trip just for one day and leaves the rat in charge of the sugarcane field. The rat hears a noise coming from inside one of the stalks and decides to further investigate by biting into it with her many teeth. The rat discovers a tiny worm and a new taste for the sugarcane. The rat ends up biting into every single stalk. When God returns, he's furious. His whole field is ruined. He punishes the rat by taking out her many teeth and ramming 2 sticks of wood into her mouth, becoming the rat's incisor teeth. God also lowers the rat in a basket down to Kosrae, where the rat decided to reside on Utunenea. Many years later, the rat ends up having a beautiful human-looking daughter. The daughter catches the king of Kosrae's eye, and they end up marrying, creating what is known as the rat clan of Kosrae. In Vanuatu, the story of how the rat got its tail begins in the forest, where seabirds are making a canoe. The rat overhears their need for a navigator and threatens to break the canoe if they do not let the rat aboard. Out at sea, an argument ensues, and the rat attempts to bite a starling. The kingfisher of the seabirds shoves its beak through the hull of the canoe, and all the birds fly away, leaving the rat to drown. An octopus swims by and hears the rat's cries for help. The octopus allows the rat to climb onto its slimy back and takes the rat to the nearest shore. The rat begins insulting the octopus on its scent, texture, and how it swam, prompting the octopus to whack a stick into the back of the rat. The rat retaliated and threw black ash from an old fire at the octopus. This explains why the rat now has a long spiky tail and the octopus has a black stain on the top of its head. Another story from Vanuatu is called The Thieves Who Helped Invent the Canoe. In this story, rats had built tiny rafts to travel to and from Vau, which was an uninhabited island. The rats would travel at night and sneak into the homes of Malikolo. The people of the village began noticing their foods, such as taro, yams, and rockfish missing. The rats even stole their toes. Eventually, a guard was able to chase down a rat to its tiny raft, but it was able to flee back to Vau. The chief ordered his men to go to the rat island, so they made a big and strong canoe and launched an attack. All the rats drowned, and the people of the village celebrated with a feast and continued to use their canoes to travel between islands. In Hawaii, the Polynesian rat is known as Iole in Olelo, Hawaii, and may be considered an aumakua or deified ancestor to some. Many Hawaiian terms revolve around Iole, such as haumaka Iole, which is a term given to elderly people that describes the blurriness of their eyes, similar to a rat's eyes. Piko pau Iole, which is the rat-stolen umbilical cord believed to make one a chronic thief in life, and papa Iole, which is a type of haircut said to be given to Piko Ika, the demigod who can appear as a rat. There are many different versions of the story of Pikoi, but in each version, he was known as the legendary rat shooter who was able to beat Mainele, the rat shooting champion. Rat shooting with a bow and arrow, or pana Iole, is mentioned in the Kumulipo as a favorite sport of chiefs and the Mo'olelo of David Malo as a minor amusement. Nevertheless, Iole were recognized as detrimental to the crops, migrating from the uplands to the shore, destroying any plant in sight. In New Zealand, the Kiore, or R. excellens, is documented in many ethnographic accounts of the Maori people through recordings of oral history. One story revolves around the departure from Hawaii, which is the ancestral homeland. The canoe, called Aotea, was used for this departure and carried aboard the Kiore, Australasian swamp hen, sweet potato, and karaka tree seeds. Other tribal traditions refer to this canoe as horuta and note the trapping and cooking techniques of the Kiore, suggesting the rat was used as a food source, possibly during annual harvesting trips to Titi Islands. Due to the introduction of Kiore by their ancestors, most New Zealand Maori consider Kiore a toanga, or treasure. However, the indigenous people of southern New Zealand, Rakiura Maori, consider R. excellens a pest because of their devastating impact on the abundance of titi, or mutton bird, which are the young of city shearwaters. Harvesting titi is a culturally significant custom and a source of spiritual inspiration and income. Some parts of New Zealand have already undergone eradication efforts to support the conservation and survival of native bird populations. However, since they are also regarded as culturally significant, sanctuaries in certain areas have also been implemented. The Polynesian rat is an introduced species that is extremely invasive to islands in the Pacific. R. excellens negatively impacts humans' health through the spread of the diseases such as bubonic plague, rat lungworm, and leptospirosis. Additionally, the Polynesian rat can spread diseases like toxoplasmosis to native animals like the nene and Hawaiian monk seal, and interfere with the reforestation of native plants. In the assessment of the health of native lands, it is crucial to conduct pono science, meaning a respectful, community-based approach where cultural values are heard and appreciated. Although the Polynesian rat is known to spread disease, devastate crops, and depredate native bird populations, the rat can also be found in the histories and stories of many cultures. Some communities, such as those in New Zealand, advocate for the presence of the Polynesian rat that accompanied their ancestors, whereas the majority on other islands, such as Hawaii, do not oppose control or further eradication efforts. A deeper understanding of the human health impacts and cultural aspects of the Polynesian rat are essential for developing native conservation efforts and implementing invasive management strategies. Thank you for listening. Questions?
How were the rats eradicated?
Rodenticides. So they would use bait stations and kill them that way.
Thank you.
So this overview that you put together, you did a lot of the research on this, right? How is your organization, because your organization is the USDA National Wildlife Research Center, so how are they going to use this information? Do you have a sense of that?
I know that they're allowing the pharmacy school here to use their lab to further investigate rat lungworm disease and see what treatments would work there. So there's that. They also work with a lot of other invasive species, such as the mongoose. So, yeah.
Cool. Great. Anybody else?
Of the diseases you talked about, which one do you think is the most serious threat to plants, animals, or humans?
I don't know. I wouldn't rank them just because lots of people have been affected by each one. I think the bubonic plague definitely isn't as prevalent as it was back then, but toxoplasmosis is definitely being spoken of a lot, as well as rat lungworm disease, especially here where, lots of slugs, like what Jess was saying with the, I forget what snail it was called, but that one with the pink eggs, that one carries the rat lungworm and a lot of other hosts as well, so, yeah.
I have a quick question for you. How did you find out about some of those stories, especially from like Vanuatu and Kosorai, like all those areas? Like that's really cool. I'm just curious, like how'd you even like find that information?
There was this really great book. I think it's called The Pacific Island Myths and Legends, and I got a lot of information from there. As well as Lennon helped me with the Hawaiian cultural aspects section. Yeah.
Mahalo.
Aloha, everyone. My name is Skylar Chong. My internship this summer was with Kumuola Marine Science Education Center down in Keaukaha, like a lot of other people's. We've been in Keaukaha a lot. And my project is titled Effects of Aeration on Organic Matter Decomposition Rates in the Local I'a of Waiahole. So that's kind of long. I think I can use this. Yeah. For those of you who don't know what local i'a are, local means pond, i'a means fish, so they're fish ponds, they're native Hawaiian fish ponds. And they represent this aquaculture practice found throughout Hawaii that was practiced for over 800 years as a way of providing this sustainable source of fish to the people of Hawaii. Recently, they haven't been doing so well. With Western contact, a lot of these practices sort of went under for a while, but then in the 70s, they sort of been starting this process of revitalization. So I work at 3 different local i'a found on the island of Hawaii. And then Keaukaha, for those of you who don't know, we've been in Keaukaha a lot, like I said. It's right over there in the Moku of Hilo. And then if you zoom in, you can see that's in the Ahupua'a of Waiakea. And then actually, the local i'a that I worked at are in this Ilukupono of Honohononui. And I have this map that is a composite map, so there's 2 maps, so some of the names are upside down, some of them are right side up. But basically, the top is the ocean, the bottom is the land, and so that road that you see coming out from the left-hand side is the road that you drive down to go to Keaukaha, if you know the area. And there are 3, like I said, 3 local i'a that Kumuola manages. They are right there, so Waiahole, Kapalaho, and then Waiopio. And yeah, that sort of box outlines the property line for Kumuola. Kumuola is run through Kamehameha schools, so we do function primarily as an educational site. The goal is to get keiki back out onto the Aina, get them involved in this work, and sort of ensure that story progresses into the future. We also function as a place that's trying to restore the fish pond, right? We want to be able to produce fish from our ponds, and we want to be able to provide food for our communities, right? To get that sustainability back into our food systems. But that's not easy, right? These spaces have been unmanaged for a long, long time, and so there are problems with invasive species, sea level rise, reduced water flow. The road that I was just talking about actually was built on top of one of the kuapa, that road, or the wall of the fish pond, so that's a big issue. But yeah, so there are a lot of issues. I wanted my project to focus on one of those, sort of address some of those things that might help us create a better fish pond system. And so my mentor and I decided to look at sediment in the ponds. And a little bit of sediment in the pond is a good thing. In fact, we know that this area is known for its sediments, more specifically the smells that come from our sediments. Because if you've ever been there, you know they don't smell the best. So if you look at some of those place names, Hono Hono Nui, Hono can be a scent. Hono Hono Nui means that there's plenty of that scent. Kapalaho can be interpreted a little, there are different interpretations, but if you break it up as ka, palaho, palaho is another type of smell. And then hauna, which is one of the names for one of the other ponds in our system, just means it's a bad smell. You don't want to smell hauna. So yeah, a little bit of sediment is good, and we actually know from our names, we say hemana ko kainoa, we say our names have power, and so we know that these spaces are known for the sediment. But today, the sediment's a problem, and it's a problem because we have so much of it. If you look at that picture on the left, that is a little pond, it's an offshoot of Waiahole, and then there's about 6 inches of sediment on the top, or 6 inches of water on the top, and then 4 to 5 feet of sediment on the bottom. So it's filled with sediment, and it's been sitting there. That place was uncovered in 2018, so that sediment's been sitting there, no change. It's just been filling up our ponds. And the sediment's bad for a couple of reasons. Oh, well, actually, wait. How does the sediment get there? Why do we have so much accumulation? It's actually because of that grass you see in the background. It's called California grass. It's an invasive species. It's one of the main invasive species that we have to deal with in our ponds. And it covers the ponds. It'll grow out onto the water. It's tolerant to salinity, to different salinity levels, so it grows out on its pond. It forms these really dense thickets, and then as it grows, it decays, it drops all this stuff, this organic matter, into the water, and that slowly becomes sediment. So we have these stands of these thickets of grass that have been there for decades, build up a bunch of sediment, and then when we remove that grass, which is a lot of the work that we do at the local IA, when we remove that grass, we're left with all this sediment, and it's bad for a couple reasons. One, it's covering the rocks, and so we need our rocks to be exposed for limu to grow, for algae to grow, because limu is what feeds our fish. And so if we have sediment covering all our rocks, we don't have limu, we can't feed our fish. And that's not great if you're a fish pond. The other reason it's bad is because the sediment itself actually produces harmful compounds and chemicals. I'm going to go a little bit into why, and it has to do with the scope of my project, so pay attention. When you have, so sediment has this general profile to it. There's water on top, and then there's a little bit at the top that is able to receive oxygen from the water. And then the majority of the sediment at the bottom, there's no oxygen in it. And what that means is that bacteria and the microbes that are in the sediment, who are trying to break it down, digest all that organic matter that's in the sediment. Remember, organic matter is coming from the grass, so there's a lot of it in there. They're trying to get rid of that. So they're trying to break it down, they're doing their thing. And in that oxic layer, they can use oxygen to break it down, which is generally thought of as a good thing, partially because it produces carbon dioxide, less harmful than methane or dihydrogen sulfide. But when they don't have oxygen, then they have to resort to other things. They use things like iron, sulfate, even carbon, and then they're producing those harmful chemicals, methane and dihydrogen sulfide, like I mentioned. So it's not great for our pond, and that's why we are wanting to get rid of it. For those two reasons, limu, harmful chemicals. So we want to get out of our ponds. But how do you do that? It's a real challenge. What we do oftentimes is we use our hands, we use our buckets, like that picture, and it takes a long time. It's also hard because you can't get all the sediment out, right? You have water and then sediment. If you stick a bucket down into water, try to pick up sediment, you're just going to get water, right? You're not going to get any sediment. So it's really hard. It's hard work. My shoulders were sore for a couple days there. But then you could think of like trying to use a dredge. We have a lot of debris in our sediment, so it might clog up a dredge. You also don't want really heavy machinery on this aina because it's super saturated with water. And if something falls into the pond, that's game over. That's not good. So there's this question of how do we get rid of our sediment? And what my mentor and I came up with was sort of using this this concept from before where we have oxic and anoxic sediment. And generally, the thought is that that aerobic process of decomposition, of those bacteria digesting that organic matter, that's faster. Generally, we generally think, that was the hypothesis at least, that that's faster than when the bacteria have no oxygen. So in those anoxic things, they're producing harmful chemicals, and it's actually going slower. So our thought was, well, what if we can use those bacteria to do our job for us? What if we can use them to get the sediment out? And specifically, well, we want to use those oxic guys, those aerobic bacteria and microbes. So we were like, what if we put an air pump? What if we just stick an air pump down there, aerate that sediment? Then they can use oxygen, which is faster, and break it down for us. So that was the thought. And in my project, basically what I did was I got a bunch of sediment from the pond. I put it into these 3-inch diameter PVC pipes, and then I left some of them sit, and then I aerated other ones. So I pumped air in through these air stones at the bottom using an aquarium pump, attached those there, and then aerated some of them. And the setup for the experiment looked like this, where I had no aeration, so that was the control, 12 hours aerated a day, and then 24 hours aerated a day. And that rationale for having the 12-hour in there sort of has to do with, well, if you think about applying it to the pond, if I can use a solar panel to power my pump, that's more efficient, a better use of energy than if I'm having to use a battery to power that pump for 24 hours a day. So if I can get away with aerating the sediment for 12 hours a day, that's a plus for me. So that's why we had those 3 instead of just the no aeration and 24. So that was the original thing. I was going to take samples from each tube every 3 days and then sort of map out that change in % organic matter. And what we're expecting to see, based off our original hypothesis, that aerobic respiration is faster, is that the 24-hour group would have less organic matter in it than the air, right? And so then it forms a plug, and then when you turn the pump back on, that sort of pushes all the sediment out, and 3 of our 12-hour treatments or replicates overflowed. So we couldn't use that data anymore because there was no sediment in them. So we had to throw 3 of those out. Another thing that we saw after 18 days in the experiment, so 18 days of collecting samples from each of those, was that there was a lot of fluctuation and no real difference between each of the groups. So we were kind of bummed, thinking like, what could be happening? And we were like, well, maybe it's our sampling method. So on the 22nd day, instead of doing a core sample like I was before to get samples, I shook up all the tubes to homogenize them, took each of one and shook them up a lot. And then I took 3 samples from each one and had those analyzed. Again, we're excluding those ones that overflowed. But basically, then we compared the initial versus the final, because at the initial, they're supposed to be all the same, right? So yeah, that's what we did. The results, remember, if we're thinking these are aerated, those aren't, we want to see, or we're thinking that we're going to see less organic matter in the 24 hours. What we actually saw was something like this. So you have your % organic matter on that y-axis, and then the treatment groups on the x-axis. That initial is around 52%, which is really high. We have a lot of organic matter in our sediment. But then you look at the control, that was going down in organic matter, but the 24-hour, the 12-hour, not really much change. 24-hour, it went up in % organic matter. Opposite of what we were expecting, right? That's weird. And you can also look at it like this, where that 0 line is the initial, and then the bars represent % change. So in your control, you're going down. 24-hour, you're going up. 12-hour, you're going a little bit down, but not too much. So what's going on there? One of the things we also wanted to test was nitrogen. Nitrogen is also a limiting factor in decomposition. So maybe those ones in the 24-hour just didn't have any nitrogen. They couldn't do anything. What we found was something like this. And you look at CN ratio, it's carbon and nitrogen ratio. And our carbon-nitrogen ratios were actually pretty low, which is a good thing. That means we have nitrogen in our sediment. For reference, grass clippings are anywhere from 12 to 25 in their carbon-nitrogen ratio, and those are used for fertilizer, right? So we have a lot of nitrogen. Those bacteria should be able to use nitrogen to decompose that organic matter, but they're not. That % organic matter is going up, like we saw there. So we're still like, what happened? What we know is that the nitrogen present, right? We know that organic matter is percent organic matter is increasing, so we're either reducing our inorganics or increasing our organics, right? And then we also know that sediment is changing. You could tell from just how it smelled. Remember I said those sediments are smelly. Those ones that were aerated didn't have any smell at all. They also look different. So we know that they're changing, but there were things, but like our percent organic matter is not telling the full story, right? So what we might have missed, well, we might have not had the bacteria we needed in there. Maybe they didn't recruit in the first place. Maybe they needed more time. We also might have missed the total volume or mass of the sediment, because maybe we're just measuring percent organic matter, right? We're not measuring the whole level of sediment in there. So maybe they broke down a bunch of sediment and disappeared, but that ratio is off. And the third thing, which is my sort of primary hypothesis for what happened, is these things called mineral associations, where different minerals can actually inhibit decomposition. And one of those things is oxidized iron. And so when you expose iron to oxygen, it can actually bind to the organic matter and then prevent it from decomposing. And when you look at our samples, actually, you see that the ones that were aerated had an orange color. So orange generally could be iron. So maybe we were aerating things, putting iron in there, and then that was preventing our decomposition and throwing off our numbers. So maybe that's what happened. What's next? It's the application of this is still unclear. Right now, I wouldn't say that we should go throw a pump in the pond, because we don't know what would happen. Maybe just not at Waiahole, because all the sediments in these spaces are really different. But mainly, it needs more research, right? We don't really know if this method would work. But regardless, it's adding to our understanding just of how our systems work, how these biological and physical processes in our local EO work. And all of that works to sort of help us take care of these places and help our local EO thrive. But yeah, that was my presentation. I want to acknowledge my mentors, everyone who helped out. A lot of hands went into this. So yeah, thank you.
Aloha mai kākou. Mahalo everybody for being here. My name is Hawai'i Agiar, and this summer I had the immense privilege of being able to work with the Mauna Kea Watershed Alliance. And the Mauna Kea Watershed Alliance is a part of the Hawaii Association of Watershed Partnerships, and is one of 10 watershed partnerships across Hawaii. Through the Mauna Kea Watershed Alliance, I worked under the mentorship of Cheyenne Hiapo Perry. And this summer, the project that we worked on was the KBC Nurse Trees project in the Kanakaleo Bird Corridor. This project was spearheaded by Stephanie Jeleniak, who is a research ecologist with USGS, and is currently being continued by Amberly Pigao, who is a graduate student in the TCBES program here at the University of Hawaii at Hilo. The KBC Nurse Trees project not only aims to find the best method for native plant reforestation through the use of native nurse trees, but also is creating a more useful partnership between researchers and land managers. This is in an effort to better inform management strategies by getting information directly from researchers to managers. And none of this would be possible without the partnerships of USGS, the University of Hawaii at Hilo, the Department of Hawaiian Homelands, and of course, the Mauna Kea Watershed Alliance. And this was all funded by the Pacific Islands Climate Adaptation Science Center. Now, I know that's a lot of information to be thrown at all at once, so let's start from the beginning. What exactly is a nurse tree? A nurse tree is an old growth or adult plant that, due to its close proximity with seedlings of another species, has a beneficial effect on them. And the nurse trees that we chose to use in this study were Mawane, or Sophora chrysophylla, and Koa, or Acacia koa. And these nurse trees were tested against control plots that look like this. They were plots out in the open and surrounded by grass. So, why is this project important? Well, Hawaii's native forests are among the world's top biological treasures.
And they are certainly treasures to us, I believe, whether that be culturally, spiritually, emotionally, historically. They also provide food, shelter, biodiversity, protection from flooding, clean air, and so much more. So if we can do anything to maintain, preserve, and perpetuate these forests, I believe that we should. Which brings us to our project area. This is the Kanaka-Leonui Bird Corridor, or a map of the Kanaka-Leonui Bird Corridor, or KBC. And as you can see here, by the red circles and blue stars, this area here is our upper elevation site, which we called Kanaka-Leonui, and here is our lower elevation site, which we called Mana Road. 500 total plants were planted across both sites. 200 plants were planted at the upper site at Kanaka-Leonui, with 2 treatments being Control and Mamane, and 300 plants were planted at the lower site at Mana Road, with 3 treatments, Control, Mamane, and Koa. 5 different native species were used in this testing: A'ali'i, or Doronea viscosa, Aveoveo, or Chenopodium oahuense, Mamane, or Sophora chrysophylla, Na'e, or Dubaudia arborea, and Pavale, or Rumex giganteus. There were, if you can see here, the nurse plant setup, this is how we collected our data. In the data, the colorful dots here represent each plant. So there were 2 plants of every species planted within a plot, and these blue rectangle shapes here represent the soil probes that we used. And this data was taken a little over 5 weeks after the plants were put in the ground. And with this method of data, we tested for overall plant survival. And for the Kanaka-Leonui site, I used a two-tailed t-test, and for the Mana Road site, I used a Tukey's test. And on the y-axis for both of the graphs is the percent of survival, and on the x-axis is the treatment. So as you can see, at the Kanaka-Leonui site, there is about a 30 to 35% difference in the overall survival of plants. And here at the Mana Road site, there's about a 20 to 25% difference between the control and the nurse trees, with Koa pulling ahead of Mamane just slightly. And this is important because it essentially tells us what we had expected, that these nurse trees are doing their jobs. They're protecting these seedlings, and they're yielding more survival than they would if they were planted in the control plots. We also tested for overall species, or individual species survival at both sites. And for these, I used a one-way ANOVA. On the y-axis is the, again, percent of survival, and on the x-axis are the individual species. So as you can see, Na'e did the worst across both sites, but that is also due to the fact that they were planted the least. Actually, Na'e, many of the plants didn't even make it into the ground. The 4 other species, however, yielded similar survival, although Aveoveo did pull forward at both sites. And this is important because knowing that Aveoveo did the best, so the 2 major challenges that we have in KBC are their harsh microclimate and grass competition. So knowing that Aveoveo can survive in that, with those challenges, it's known now that we can use Aveoveo for preliminary or early reforestation. And lastly, we compared overall survival between the Kanaka-Leonui site, the upper elevation, and the Mana Road site, the lower elevation. And the reason why the histograph looks the way that it does is because when I collected the data and put it into a spreadsheet, due to the fact that there are 2 plants per every species in every plot, 0 here meant that both plants per a species was found dead. 50 meant that 1 was found dead, and 100 meant that both were found alive. So it is evident here that Mana Road definitely yielded a higher survival, but that could also be due to the fact that there were just plainly more plants planted at Mana Road. So amidst me participating in this project and helping out, I was also able to engage in things like environmental education, which is a part of what some of the Mauna Kea watershed outreach. So this is me and my mentor Cheyenne doing environmental education with Kohala Middle School during a summer program. This is at Pu'uhuluhulu. I was also able to do a lot of outplanting, and this was a lot of fun. Here is me. We were doing outplanting of ohia in the Hakalau Forest at Hakalau Refuge. And we were able to outplant at the Waipahoehoe unit in Pi'ihonua. I also learned a lot. I learned a lot about seed propagation and seed collection. This is Shane. He is one of the members of the Mikwa field crew, holding up bags of aveo seeds that we collected that day. And it was raining, so it was kind of a struggle, but it was a lot of fun. And here, these are koa seedlings that I transplanted here into these dibble sticks from this flatbed. I also learned more about fencing and fence management. This is a photo of me and another Mikwa field crew member. We did fence management and fence surveying around the Waipahoehoe unit. And lastly, I made a lot of friends and made a lot of connections and associates. It was really nice to have these connections and meet like-minded people. I really appreciate the time that I had here and spent on the Mauna. It taught me a lot, I think, about how I connect to my culture and how I connect to myself. I'm very grateful for my time, and I'm even more grateful for the people who provided that to me, including I want to give a big mahalo to the Mikwa field crew. They were really awesome to me, very kind, and very supportive. I also want to give a big mahalo, obviously, to the partners that made this possible, the Department of Hawaiian Homelands, University of Hawaii at Hilo, the Pacific Islands Climate Adaptation Center, Kamehameha Schools, USGS, and of course, Mauna Kea. And the last and the biggest mahalo goes to PIPES, because without them, none of us would be here. I would not have made the friends that I did this summer. I would not have learned the things that I did this summer. And I will never forget my experience. So mahalo nui for your time. Mahalo nui for listening. If you have any questions, please feel free to ask. Oh, and a big shout out to Becky Ostertag for my grabs.
I just had a question. Did the recent fire on Mauna Road affect your sites, or what did the vegetation that got burnt look like? Do you know?
I haven't seen the recent fire, but I'm pretty sure. Actually, I won't. I'm unsure if if KBC was affected at all. I assume it wasn't because it's very high in elevation, but I haven't seen any of the burn sites. Okay.
Okay.
Kau'i, was there any specific reason that you guys used koa and mamane for your nurse trees?
Okay. Well, first of all, the biggest reason is they're very common overstory trees at that elevation, at the mid and higher elevation. And they're also big nitrogen fixers, so they provide the seedlings underneath them with that extra, essential fertilization.
So you were talking about the bird corridor. Do you know why it's called that? Is it connecting something?
So part of the reason why the reforestation is happening is also due to the fact that they want to provide native birds with a ladder to go up the mountain, considering that climate change is, making the mosquito line go higher, and that's affecting the birds. Obviously, you know this. So that's part of the reason why we're doing this study, is also to see what's best for reforestation to help those birds out.
It's really exciting to see this project, Kau'i, and see your work with Cheyenne. And I think it's a neat example in which Cheyenne was an intern a really long time ago, I think 1998. And then Mina, that was his project in 2008. So as they were starting in the original research that was at Kanakaleo Nui, that bird corridor, because of the break between the forest and then going up up higher. So yes, mahalo. I really appreciate everything you had. So that was all. So thank you.
Next presenter, Dylan A'ali'i Kelling. We're really excited to share Dylan's project. He worked this summer with ASM Affiliates, who is a new partner this year that we worked with. So he has some really interesting, he did a really interesting project and has some really interesting information to share. So without further ado, I'll let Dylan give his presentation.
Can you guys hear me okay? Okay. Aloha mai kākou. My name is Dylan A'ali'i Kelling. And this summer, I was under the mentorship of Loke'alani Brandt and the ASM Affiliates Agency to learn more about Land Commission Awards and how we can utilize these documents in future land management. And yeah, as somebody who was raised in Kuleana lands, I knew a little to nothing about these unique land parcels. So this was a great opportunity for me to learn more about where I come from and the things that went into this project. So today I'd like to share with you some of my findings on Novaike Kuleana investigating Mahele documents to further assess its applicability in modern day restoration efforts. Okay. So here's an overview of my presentation. I kind of wanted to talk a little bit about the background of the Mahele Aina and how that played a big role in Kuleana lands. And then I'd like to talk about how Kuleana lands and its transition into fee simple titles. Then introduce my field of study, which was Honuapo Ka'u, and then talk about my methods, results, and discussion. So with the influx of foreign population within the Hawaiian kingdom and the constant pressure from outside influences, King Kamehameha Ekolu and his men realized that the Hawaiian economy could not move forward under the old feudal-like system. Therefore, in the year 1848, the king, along with 240 of his highest-ranking chiefs, divided out the land one last time in an act known as the Mahele Aina. Within the Mahele Aina, there were 3 classes of people having each 1/3 vested rights within the land. And this included the Mo'i, or the king, the Konohiki and Ali'i, or the ruling chiefs, and the Maka'inana, or commoners. As well as there were 3 categories of land divided, which were crown lands, handpicked by the king, Konohiki lands reserved by the Konohiki, and government lands, which were lands surrendered by the Konohiki and Mo'i during this time. And as the kings and chiefs began to make claims for the land, this led the Maka'inana to question where their 1/3 vested rights were. Furthermore, the lands awarded to the king and Konohiki were all subject to rights of native tenants. Therefore, on August 6, 1850, the Kuleana Act was passed, authorizing the Land Commission to award fee simple titles to native tenants who either cultivated or resided on a land in the Crown, government, or Konohiki lands, and these lands would be known as Kuleana lands. And fun fact, tomorrow would be the 171st anniversary of that the Kuleana Act was passed. So as a native tenant, the process to make claims, you would begin by a handwritten letter to the Land Commission Award, which is also known as the Native Register, and this would be sent to Oahu via steamboat. And then next, you would need a native or foreign testimony, and this would usually be done by a neighbor or somebody who can vouch for you that you have indeed claimed these lands or had actively cultivated these lands, and then you would wait for approval. Okay. So here's my field of study. My field of study was located on the southern eastern side of Hawai'i Island, within the district of Ka'u in the Ahupua'a of Honuapo. Also known as Whittington Beach Park, the coastal community is a sheltered bay that has an estuarine local i'a and a wharf used in 1883 by merchant ships to transport raw sugar during the plantation days. While the inland was more known for its rich soil, its optimal farming, and adequate rainfall. For this project, the Kuleana land parcels were analyzed within the Ahupua'a of Honuapo. Here is a little site description. I analyzed all these land parcels in blue. That's the whole Ahupua'a, which is north from Ki'oloku and south of Hiona'a, in which 24 land parcels were recorded and 21 claimants were found, meaning that some of the claimants within the Ahupua'a had more land parcels than others. And each of these land parcels are divided with their distinct boundaries and a unique Land Commission Award number. Okay. So my research question aims towards the coastal community within Honuapo, and my research questions were, what can we learn about the resources and community within Honuapo from mid-19th century Mahele documents, specifically the cultural practices coastally and agriculturally, and how these information can be applied in modern day restoration efforts. For this project, the Land Commission Awards were analyzed using the Kipuka database, which is a geographical information system, or GIS, that encompasses mapping and Hawaiian teachings and history. And within each Kuleana land parcel, they were awarded a Native Register in A, Native Informed Testimony in B, Land Commission Award in C, and a Royal Patent Grant in D. As you can see above, this is an example of an awardee, Lili Kalani, who was a claimant within Honuapo. And majority of these documents were written in longhand cursive and varying legibility. Each of these documents were all analyzed, transcribed, and translated from the Hawaiian language to English in which the data was collected. As a result of the Mahele of 1848, the roughly 2,000-acre Ahupua'a of Honuapo was awarded as konohiki lands to William Charles Lunalilo. As for the native tenants within the Ahupua'a, the smallest awarded lands was 0.3 acres, while the largest was 11 acres, with an average of 4.5 acres per claimant, and a total acreage for native tenants of 97.42. These lands were awarded within the years of 1853 and 1887, and majority of these claimants within the Ahupua'a had to wait a very long time for them to be granted these lands. The shortest time awarded was 17 years, while the longest was 48, with an average of 34 years being awarded. And many of these claimants within the Ahupua'a either would not survive long enough for that time and would have to pass on the land to a successor. However, if there was no successor, then the land would be redistributed. As for the supported lifestyles within the Ahupua'a, I found that agriculturally, a lot of the claimants had kihapai farming, which is a dry land method of farming in a cultivated patch or small farm. And within the kihapai farming, they had cultigens that went for food, tools, or clothing. So food such as kalo, uala, niu, mai'a, and ko were all used and found. Tools were hau, and the vauke was used to make kapa or clothing. And then what was interesting that I found was that paka or baka was recorded, which is the tobacco. And what's interesting about that is it was a plant that wasn't necessarily native to Hawaii, but it was introduced. So we can see that these natives of Hawaii have been introduced to these plants and are actively doing something with these plants. And then the naku was a natural resource grown within the local i'a. 36% of the local claimants within the Ahupua'a own poho pa'akai or salt beds, and they were usually generally within the coastal part section. And then there was only one person within the whole Ahupua'a who claimed the local i'a, which was mentioned earlier, Lili Kalani. And within his native testimony, he mentions having a kuleana loko that he worked with along with his men every Tuesday. And within the local i'a was called ama or mullet, and ava or milkfish was cultivated. So here's some of the Ili Aina names. So Ili Aina is the subdivision below the Ahupua'a. So it'd be Mokupuni, Moku, Ahupua'a, and then Ili Aina. So within this Ahupua'a, there was 17 different Ili Aina locations recorded, many of which could not be pinpointed to their exact location because there was not enough information within the testimonies to do so. However, here are some of the results of the names recorded. So in examining the Kuleana land documents in the coastal community of Honuapo, there were several cultural practices that were identified, which included agriculture, aquaculture, and salt production. The pohopa'akai, or salt beds, can still be seen to today on the coastal community. And what was interesting about these salt beds were that they weren't just used for salt, but they were also used for making hauna, or chum, for bait and fishing, as well as dyeing of kapa, because these salt beds varied in depth and sizes, so they could use it for multiple things. As for the local i'a, or the fish pond within the ahupua'a, it still can be seen till today. Although the inlet structure and shape has changed, I feel like there are restoration efforts being put into the process. Or as for the change of lands within the ahupua'a, the coastal community is now protected by a nonprofit organization called Ohana o Honuapo, and their mission is to protect the natural resources within the park and educate the community on the importance of the bay. Other changes in land include plantations. So this was a big one that I wasn't really able to look into because these native testimonies and stuff came a little before the plantations, so I didn't really have enough information to talk about these plantations. Therefore, future studies should analyze the many documents of the past, such as Hawaiian newspapers, passages from historians and scholars who talk about changes in land more recently and how these plantations can be applied in land management. Another thing that could be used for future studies is to actually pinpoint those ili'aina locations, because I don't think they're actually being used, and not a lot of people know about these ili'aina locations, meaning that if they're not used, they're gonna be dead, they're not gonna be relevant, I guess. But at one point in time, they were used every day, like Target or Walmart. So I feel like future studies should utilize these locations to locate where Hanuonalani is or Kepauole is, and really revitalize these Hawaiian names. As someone who was raised on Kuleana lands in the ili of Ioleka'a He'eia Ko'olau Oahu, I'm grateful for this opportunity to learn more about the functionality of these lands 180 years ago and its applicability in shaping our future. It is essential to know where I come from and the stories that have resonated with the past. This is my ka'au through the lens of finding applicabilities of traditional knowledge in a modern context. Okay. This study would not have been possible without all of these awesome sponsors. Yeah, mahalo ya oko. Mahalo, Nui. Yeah, do you guys have any questions?
Hi. Besides documents, did you handle any artifacts? And if so, what were they used for?
Yes, that is a good question. I didn't specifically handle artifacts within the ahupua'a of Honuapo, but I did get to do a little bit of cleaning of artifacts within the ahupua'a of Papa'iko, and it was a bunch of bottles from the 1800s that I got to clean, and it was a lot of work. It was just a lot of shards and stuff. So, but yeah, I got to clean some artifacts. Mm-hmm.
Thank you.
Yeah, thank you.
What was your favorite part about participating in this project?
Ooh, yeah, that is also a good question. I think one of my favorite parts was getting to relate where I come from to a different place, a community that I was not really familiar with, because I did have the opportunity to kind of look at my Kuleana lands and where I come from. And it was really interesting to see who claimed the Kuleana lands before me, which was a tenant named Kekua with the Land Commission Award number 4221. And it's kind of cool how he describes the land before and how he mentions the stream that runs through the property and the different lo'i he cultivated. So yeah, it was kind of just cool relating two stories together. Yeah.
Mahalo.
Yeah, mahalo.
Aloha.
Aloha.
Because you had to deal with a lot of Hawaiian language documents, can you describe how important going to a Hawaiian charter school was to your work and the importance of?
Yes, definitely. Yeah, I think this was a big part of my project. Like, without understanding or knowing the Hawaiian language, or without going to a Hawaiian charter school, I don't think I would have been able to translate or transcribe these documents, because majority, if not all of it, was in Hawaiian, and all of it was handwritten. So it was really hard for me at first, but luckily, because I had that Hawaiian language background, I was able to push through and, yeah, transcribe, translate. And mahalo for your question. Mahalo nui.
Aloha everyone, my name is Amy Patterson, and before we get started, I'd first like to thank you all for coming to the PIPES 2021 Symposium and taking the time to listen to my presentation. This summer, I've been working with the Multi-Scale Environmental Graphical Analysis Lab, or the MEGA Lab, to analyze temporal trends in disease and growth in Parietes lobatic corals at Laihala, Hawaii. So coral reefs are extremely important to the entire marine ecosystem because their productivity supports more species per unit than any other marine system. They also act to protect coastlines from storms and erosion, which can devastate coastal communities. They also indirectly support the economy through the jobs, recreation, and food they provide. In this economic realm, coral reefs are estimated to have a net economic value of tens of billions of dollars. Therefore, the health of reefs directly correlates to the health of the entire marine system, coastal populations, and the economy, making it especially important to characterize coral health and growth. So why is coral research important? Well, long story short, coral reefs are disappearing. Coral disease is a significant contributor to this reef decline, accounting for nearly 6% of global loss. Disease outbreaks have also been the primary cause of the loss of reef ecosystem function in places like the Caribbean. And since we want to keep Hawaiian reefs from the same fate, it's important to track disease and its progression in coral so that we can later find solutions. Growth is also a large indication of coral health because an increasing mass means that the coral has a suitable environment and enough resources to support its physical expansion. Coral growth means that the reef is on an overall positive trajectory and has a good outlook. So, to restore healthy marine ecosystems, it's essential to understand the baseline state of corals in terms of disease outbreaks and growth rates. So for this study, I specifically chose Parietes lobata, or as it's known in Hawaiian, Pohakupuna, for this study due to its role in reef ecosystems. It appears in large colonies with mounding morphologies and exists in cream, pale brown, or green colors, as you can see in the picture above. If you've gone snorkeling anywhere on the Big Island, I'm sure that you recognize it as it is very common. It's also a reef-building coral, which provides structure for reefs and supports the growth and presence of many other types of coral and marine species. It's also a slow-growing and highly resilient coral, and these characteristics make it the backbone of reef ecosystems. So the health of this species is likely to cascade through the entire marine ecosystem, making it extremely important to characterize. The objective for this project was to use 3D modeling techniques to characterize temporal trends, or change over time, in growth and disease in Parietes lobata colonies between 2019 and 2021. Specifically, my research question was, how has coral disease and growth progressed in Parietes lobata colonies from 2019 to 2021 at Laihala on Hawai'i Island? From this question, I developed the hypothesis that disease coverage and the total surface area in single Parietes lobata colonies will increase between 2019 and 2021. I hypothesized that disease would increase given the understanding that once a coral colony has been infected by disease, it typically results in the death of the entire colony, which I expected to be reflected in my observations. Also, under the assumption that corals grow over time, I expected to see an increase in surface area between these two time points. Surveys of my Paraides lobata colonies were done at Laihala and Keaukaha. This site is unique in that it consists of a protected shallow reef environment with a natural lava rock barrier which partially separates it from the open ocean. This quality reduces factors like wave action, as well as exposing the coral colonies to similar conditions of depth, temperature, and chemicals in the water. It also makes the repeated monitoring and survey of coral colonies manageable, making this an ideal site for this type of research. So to complete my temporal analyses, or change over time, I used plots of P. lobata colonies previously surveyed and modeled by members of the MegaLab team in 2019. Then I re-identified these same colonies, created 3D models of them, and compared them to the previous models in 2019. In total, I surveyed 4 plots and analyzed 7 colonies between 2019 and 2021. This is an example of a comparison of one of the colonies from my study, with the 2019 model at the top and the 2021 model at the bottom. You can already see clear differences in color, arrangement, and algae growth. For example, in the 2021 model, we see a concentrated amount of algae in the center of the colony, while in 2019, the colony lacks any apparent algae growth. To collect my data for 3D models, I used a Canon SL2 camera housed in a waterproof Ike-like casing. I swam over each plot in a back and forth swimming pattern with an 80% overlap between each photo. I also used reference scale bars in the plots and images to later build 3D models scaled to the correct size. To create the 3D models from this data, I used the Agisoft software to perform structure from motion techniques, which is essentially a fancy way of saying creating a 3D model from a 2D image sequence. And this is done by aligning overlapping images based on shared features while applying scale bars. I also built 2.5D digital elevation models of each colony, which provide graphics on the elevation characteristics of each colony. So I know you're all probably wondering what these 3D models look like, so let's take a look. This is a plot that I used in my study. As you can see, I used 2 scale reference bars, one for elevation and one for 2 dimension. And this shows 2 colonies of Paraides lobata. After building these models, the proportion of the colony overtaken by disease was calibrated through the CoralNet program, which randomly distributes 400 points across the plot, and each point was then labeled as either diseased, healthy, or a non-coral hard substrate. The number of diseased points divided by the number of both healthy and diseased points then yielded the proportion of the amount of the colony affected by disease. Growth was calculated in the ArcGIS program, and colonies from 3D models were outlined to create shape estimates, and shape area was calculated through the program. Diseased areas were also outlined and subtracted from the total surface area of the colony to ensure that only healthy tissue was included in the measurement. So the results for disease showed an average increase of 5.05% in the proportion of diseased points. Only 2 colonies showed a decrease in the disease coverage, while 5 colonies showed an increase between the time points. The colony with the highest change in decreased presence showed a 29.53% increase in the proportional disease, and the colony with the lowest change showed a decrease of 13.1% proportionally. Although we do see trends in the averages, I ran a Mann-Whitney test, which yielded a p-value of 0.7104. And if we look at, which means that we do not see a significant difference between 2019 and 2021 in terms of disease coverage. And if we look at the data distributions, we do see a very similar mean between these 2 data sets, represented by the bold line in the box plot. In addition, we see very similar central tendencies, represented by the wide area in the colored portion of the violin plot. So because these data distributions are so similar, we do not see a significant difference between disease coverage between 2019 and 2021. Growth was measured in centimeters squared, and yearly growth rate was calculated between the years of 2019 and 2021. The results for growth indicated that there was an average decrease in colony surface area of 6.3 centimeters squared per year. Only 2 colonies showed an increase in surface area, with the highest change being 0.79 centimeters squared per year. And 5 colonies showed a decrease in surface area of healthy tissue, with the highest decrease being 11.03 centimeters squared per year. I also ran a Mann-Whitney test on these 2 sets of data, which yielded a p-value of 0.62, also showing that there is no significant difference in the size of colonies between 2019 and 2021. Similar to the disease coverage distribution, we see a very similar median in these 2 sets of data, as well as similar central tendencies. So because these 2 sets of data are so similar, we do not see a significant difference in the size of P. lobata colonies between these 2 time points. So the results show that there are trends in the data. Averages show that there was a 5.05% increase in the proportional disease coverage. And given that coral typically experiences entire colony mortality once infected by disease, this observation could be a result of the disease simply continuing to progress, because all colonies in this study were diseased in both 2019 and 2021. In addition, there was a trend seen in the decreased overall growth rate. This could be explained through the observation of an increase in disease. Since disease kills healthy coral tissue, the continuing progression leads to less healthy tissue, thus resulting in a smaller surface area. So because we do see these trends, it is important to keep monitoring and surveying these corals, because over a longer time period, we could see more of a significant change in these aspects. And from these findings, the large p-value signifies that there is no significant difference in disease coverage and the size of P. lobata colonies surveyed. This could be due to a small sample size of only 7 colonies, which simply was just not enough data to prove the trends we saw in the results. However, not seeing a significant difference is a positive result, because the reefs are not in decline, like so many of the other reefs in the world. This also speaks to P. lobata resilience, because all colonies were diseased in 2019 and 2021, and they're not showing a significant decline in health. In addition, they live in a very shallow environment. They get so shallow at times that the colonies are even exposed to the air. So the fact that they are not showing a significant decline in health is positive and speaks to their resilience. That concludes my presentation. I'd like to give a special thanks to my mentor, Dr. John Burns, Kaylee Pascoe for guiding my project, the graduate students at the Mega Lab, and the PIPES coordinators. I'd also like to thank all of the organizations that made this unforgettable experience possible. Thank you. Any questions?
Don't block that.
Hello? Okay, there we go. You folks only did about 7 coral samples. How many samples do you think that you need to actually take to get an actual accurate reading?
That's a good question. I think that at least maybe doubled would be good. However, I was limited in the amount of coral colonies that I could sample, because there were a limited amount of plots from 2019 that I could resurvey. So definitely a larger sample size of at least potentially 15 to 20 would be ideal, but however, I was limited in this study.
Thank you.
I'm just curious, since you have lots of the data and also pictures in the field, did you spend more time in the field or more in the lab getting the data?
I really only spent about 4 days in the field, maybe 2 hours at a time, just to collect my data, to re-identify the plots that were from 2019. So yeah, most of it was on the softwares, building the models, and analyzing the data.
We got a question online from, I don't know who this is from, but the question is, were there any other species at Laihala you would like to add to this study if you could repeat the work?
I think because Pilobata is it's like a reef backbone species. I would really be interested in researching corals that rely on species like Pilobata. For example, there's a species called P. mandrinus that is reliant on Pilobata, and see how these corals are responding to things like disease and growth as well.
So if you were assessing the disease by the colors of the pixel, did you have to like process the data to take into account that it was taken underwater and the water quality or depth might affect it?
Yeah, so we actually, before taking the images, we used a white balance technique with the camera to make sure that the underwater images were the correct color. And I think that it's pretty obvious when a coral is diseased and when it's regular color, because you can see there's distinct spots of discoloration, algae growth like you did see in that one plot. So yeah, we did account for that.
That's all good. Thank you. Thanks, Amy, for a great presentation. So how much of this technology, how much of this did you know before you started this internship?
Absolutely none. When I first figured out I was doing this project, I was super nervous, super intimidated, I would say, because 3D modeling sounds like a lot of computer work, and I am not a computer science person, but it was a really good challenge, and I really enjoyed every step of the way.
That's great. And so does this lead you into thinking on other things into the future? Does that open it up, what you might be interested in doing?
It definitely does. I think that even building the 3D models and like taking something that I saw in the water, producing it on my computer and like being able to move it around and see it was so interesting. And I feel like I can really see myself doing something similar in the future.
Great, thank you. Well, thank you. Let's thank Amy for a presentation. Wonderful. Well, I think that is it. Maybe we can, there we go. Thanks, Blaine. So mahalo, everybody. I didn't check my numbers on who everybody, it's been running between 30 and 40 people online. So thank you, everybody, for watching online. We're all, if you can see the room, everybody's like, kind of wiped out. But this was a really great time. And I think, see if I can get less feedback here on the mic. No. Yeah. Sorry. Oop. There we go. Hold it on the bottom. Don't hold it on the bottom. Got it. I wanted to thank all of the different people that were helping support this program. This program is supported by our partners. If there are people in the audience who are interested in partnering with us, please contact us. You can contact us at uhintern@hawaii.edu. Go online and you can contact me, Sharon Ziegler-Chong, or Devin Aguiar, or Mina Veratua. We're really excited this year just of having an internship program in general. Last year we did everything online. We did not have any groups together. This year we pulled it off by only doing PIPES on this island. We usually are across the state and often into the Pacific. And so this year what an important part has been to connect together in place. We've had to do with a lot of masks, a lot of social distancing, and just having people together. I think when we think tomorrow we're going to go, thanks to KMS schools, we're going to go back to Laihala to go back to where we started the whole thing 10 weeks ago, which will be really special. And I've just got to say that, every year, this has been my summer, every year for a really long time. And I think that every summer is really special, and so this year was definitely a special one, all of its own, all of its uniqueness, and the individuals as well as the cohort. That's always the fun part of it, is seeing individuals going from one place to another, meeting their organizations and agencies through your eyes, because we know a lot of these people, but what we're learning is a lot of things about them from our students, and creating even closer binding with those organizations through these experiences. So what we've one of the things that we challenged. Are we almost there? Yeah, we are there. One of the things we asked the interns to do is to do a Ho'omohala project in which they are giving back. What is this idea that they are giving back to the group? And so they've created a video, so that's what's on right now. Devin and Mina have been getting it on, and I think we will be going live to that. And so then after this, we'll say a mahalo to everybody and wrap it up.
Pacific Aquaculture and Coastal Resources Center. Identify, cultivate, and better understand the importance of native limu of the Hawaiian Islands. Speaking kind words and listening to the limu while helping the species thrive.
This summer, I was an intern at the Pacific Aquaculture and Coastal Resources Center, also known as PACRAC. At PACRAC, I gained knowledge of cultivating limu in controlled environments. I gave positive energy and efforts towards the work done at PACRAC as just one of many helping hands around the facility.
This summer, I worked at the local ia of Waiahole, Kapalaho, and Waiopio. I gave a little bit of blood, a lot of sweat, and I don't think any tears, but if I did, they were tears of joy. I gained so much from this experience, so much knowledge, so many lessons, but one of the more valuable things that I gained were the pilina that I formed, both with that place and with the amazing, amazing people I worked with.
I had the amazing opportunity to work in Keaukaha with TCBES graduate students and professors. I gave a lot of my sleep, but also a lot of my aloha to this project. I was gifted with lots of knowledge and a newfound enthusiasm for research.
Laihala tide pools. I gained an appreciation for corals as growing, living, and fragile animals in an ecosystem rather than inanimate mounds. I gave an ear to the coral and a mode of communication so they could tell me about their health as individual colonies and as a species.
Every second, every minute, every hour, every day, we celebrate this beautiful scenery. Like the K-E-A-U-K-A-A, that's where we stay. A-B-E-N-L-O-A.
2021 was with Bryce Masuda and Amy Durham of the Hawaii Endangered Bird Conservation Program. For my internship, I believe that I gave conservation a new lens to look through. Coming from a Hawaiian studies background, where we often look behind to move forward, and to take mo'olelo and stories as not just history, but as fact. In a field where research is done selflessly, I poured my spirit into. I hope that my example can bridge the gap between science and culture. To me, what I've gained from my internship, I can't possibly put into words. How do you thank someone for giving you a new look at your future? Who has shown you a new possibility that you've never imagined for yourself? You can't. But what I do know is I will take these lessons to heart, internalize them, and never forget them, and in time, pass them on. Mahalo ya oko no na mea vai vai.
My name is Sophia Smith, and my internship this summer was with the USGS, looking at the role ambrosia beetles might be playing in the spread of rapid ohia death. I gained a ton of experience both in the field and in the lab, which helped me realize that I do have what it takes to be a scientist, even though I was doubting myself before. I hope I left some valuable information that can be built upon for future studies.
My internship took place among the Kalapana Nui coastline in Puna, where I call home. I gave my time to document this coastline with the hope to preserve and restore our native ecosystems for future generations. Throughout my experience this summer, I gained the understanding that making and fostering relationships within my community and beyond are important to promote positive impacts to any conservation project.
Aloha, my name is Ka'epukai Goen, and I'm a proud intern of PIPES 2021. My internship took place on the beautiful Kalapana coast with Gina McGuire and Kai'ini Pu'uwai Keili'i Ho'omalu Holtz, recording coastal vegetation as well as the lemur species found off the cliffs and in the tide pools. I gave my kokua as much as possible with an open mind to an area that I am not familiar with. And what I was blessed with was the knowledge of this place that is so momono with life. It has been helping the communities of Kalapana for many generations. I gained a new perspective on how other Hawaiian communities operate and protect their coastal resources, as well as a new family. Mahalo nui loa to the Pipes Ohana.
To work down in Waipio Valley, doing lo'i restoration, traditional lo'i restoration, and one thing that I have to definitely take away from this whole experience is this one phrase or quote that stuck with me so hard is when a river jumps. When a river jumps, said by my mentor and her father and her whole family, and I'm pretty sure everybody in Waipio, but basically when a river jumps, it's suddenly diverted or changes directions due to like a falling tree or landslide. And to me, that matches real life too, because obstacles fall in our life and our path of life all the time. And we jump, and we must adapt to the changes. And I learned a lot about not just lo'i restoration or becoming a better scientist, but I also learned a lot about becoming a better person in general. And this experience has been the utmost rewarding, but also the utmost one of the biggest learning experiences, and I'm really appreciative and grateful for this opportunity. Okay.
Aia ka'u hana ma ka hua hoama ma ka ili o vai puna lau, ma ka ahu pua'a o Pa'uilo, ma ka moku o Hamakua, ma ka moku puni o Hawai'i. Loa ia u ka ike o keia wahi, a ua ha'awiaku au I ka'u manawa, ka'u hana, a me ka'u aloha. Mahalo piha ia hui mau.
Aloha, my name is Isabel. This summer I worked in Volcano in the National Park and in Keoho. This summer I learned about the history of Iliahi in Hawaii and the behind the scenes of native plant restoration efforts. Besides my time, I gave an open mind to experiencing new opportunities, learning about plants, and meeting new people.
This summer, I worked with mentor Lokelani Brand and the ASM Affiliates Agency
to learn more about Mahele documents and its applicability in modern-day restoration efforts. I gave my research to the community of Honuapo in hopes to educate and support their acts of aloha aina. Thanks to the PIPES internship, I was able to gain valuable work experience, a chance to develop and refine my skills as a future scientist, and a very strong support network.
I spent my summer on the Mauna, and that aina gave me a peace that I have never known, and I believe that I gave it love that I have never had for anything else.
Hi, this is Cole. I work at the USGS with Tim Grabowski and Michelle Shuey. I gained a lot of experience with how to do science with them. They taught me stats and how to interview, and I learned how to work on a team with Drew. Shout out to Drew. And I think now I just really have a different colored perspective on my relationship with my home. And for that, I'm really grateful. Mahalo, thank you.
I did my internship at the SDAV lab at UH Hilo, and one thing I gained from the internship was more knowledge about remote sensing, and one thing I gave was more knowledge about how ROD is affected by ungulates.
I worked in Hilo Bay from Kaipalawa landing to Pu'umaile, surveying fishermen about how COVID-19 impacted their lives and fishing practices. And while working along the stretch of coast, I gained a better understanding of the fishing activity at the sites we studied. Also, history about Hilo town and those sites. Uncles were very willing to just explain anything we were curious about. Even if we weren't curious about it, they just dropped knowledge. It was great, learned a lot. I also built relationships with some of those anglers and expanded on my relationships with the places that we frequented. And at every site we greeted people with genuine kindness and curiosity, so I like to think we gave back by sharing aloha with those guys and providing an outlet for them to convey their thoughts and experiences or anything else that was on their mind. And I also picked up all the trash I saw in an effort to give back to the places themselves.
This summer, I interned at the USDA office in Hilo under Dr. Stephen Hess and Carmen Intaki. As an intern, I gave my best efforts to produce thoroughly researched sections for the Polynesian rat paper. Through this amazing opportunity, I was able to gain technical writing skills, a deeper interest in conservation, and role models that I'll never forget.
This summer, I worked in an intricately ordered UH Hilo lab. I feel that I've really gained a greater sense of self, and I think what I have now is just a better perspective on what I want to do with my life and for other people.
Wow. That was great, you guys. That is so much fun. Thank you so much. That's a really neat project. Mahalo nui. I want to do just a really quick shout out to everybody. Mina and Devin. This is their first year. It's kind of their internship year of coordinators. They started pretty much right before the program. They've been drinking from a fire hose and treading water, and I'm sure they felt a little bit like they're, how in the world are we gonna get through this? Every day something more was thrown at them. They were so cute some days. They show up and like, I think we're pretty much caught up, and then I would like give them a list of 20 things, and they would be like, okay, we got a lot to do. So anyways, mahalo nui, let's give a hand to them. Come up here. Come on. And we stand on the corner, the shoulder of giants. There are really, really important people that are make this possible, and I'd like to give a shout out to them as well. Two of them are in this room. Becky Ostertag is in the back. Becky, thank you so much. Becky is our PI on our National Science Foundation grant and has been for several years now. And she's just such a force of that this program really couldn't continue in the same way without you. So thank you, mahalo nui. And I know she loves the spotlight, so it's like, yeah. The other person who really loves a lot of emotional gushing is Noi Punivai, who is also in the back. Mahalo nui. And Noi is our co-PI on the RU program, but has been a former coordinator, was a PIPES intern, even longer ago than Cheyenne. And just has been a heartbeat of really making this program, taking it from kind of a STEM internship program into much more, and one that's really focused on Hawaii and the Pacific Islands, and bringing all of these things together that you've been able to see throughout all these presentations. So mahalo nui noi. Yoo-hoo, go noi. There are also two other people, Lisa Canale, who works here at the University of Hawaii with the TCBS program, is our writing coordinator. She's really helped individually for each person in different ways, and she's been a really heartbeat of our support system. So mahalo nui, Lisa. And Lisa, I think, is waiting for a few papers, yeah. Definitely not least at all, Ulu Ching with here in our community, who is a former coordinator, but has been such an important part of our ohana in guiding us as well, who has joined us in various things, and we hope to see her tomorrow in our final day as well, has been a really good thing. So woot woot, thank you, Ulu, mahalo nui. We have a great ohana here of PIPES alumni. These guys now join over 700 people who have been former PIPES interns and the alumni network. And that's pretty neat that these guys now walk into that group, into a network of connections, of people who are really focused on aloha aina. And so I really mahalo everybody, and including Blaine and Raymond, who have been our IT support and making this really an awesome experience. Thank you very much. And then I'm going to hand it over to these guys to just bring it home and say mahalo. So here we go, guys. I know I'm putting you on the spot. Here, one last thing I need to hand to them here.
You can close it. You have a really good job at closing it. I just want to add two things. So like Sharon mentioned, this is our first summer doing PIPES as coordinators, being in this position. And for the past, I don't want to mention how many years, but it's been a long time since the inception of this program. And it had been ran by wahine, strong wahine, my kumus. But the baby never died, the house never burned down, and we all good. So I think my boss is happy with the product. So mahalo each and every one of you for dealing with, setbacks, and the road is always bumpy, yeah? So mahalo everybody, our homana, for, bearing with Devin and I as we go through this process. I also just want to mention that a lot of our homana have been talking about pilina, and we actually failed to address this in the beginning. So every year we have a theme. And this year's theme was, we were reflecting on our connections to ourselves, our aina, our community, our ohana. And we came up with the olelo no'ea of Pilina Vaivai, and kind of reflecting on that. So having that mana'o, we definitely have strengthened our Pilina to each other, to our ohanas, to our aina, as you all have mentioned in that video. And to ourselves, I think, we definitely strengthened our pilina to ourselves. So mahalo, 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 mahalo to everybody who made this happen, possible. And I'll give it to Devin to bring it home. Mahalo.
Mahalo, Mina. I think, yeah, Sharon let me know I can come back to work Monday, so I think we're good for the next year and all. So, but no, thank you, thank you guys. It's been a great summer. Thank you, Sharon. Thank you, Becky, Noi, Lisa, Ulu for helping us, because like I said earlier today, it's funny, like we're both former interns and now we're on this side of it and seeing the amount of work that goes into it. It's a lot, but it's very rewarding, which I'm also seeing as well. So I'm glad to partake in that. Yeah, I guess just some words of advice. Yeah, like Sharon said, you guys are PIPES alumni. You joined this big web network, so use that to your advantage. Network, make those connections. And we're also here to support you guys as well, even afterwards. So let us know how we can support you guys moving forward. I don't really have a script or anything for this, but a video, guys. Especially if I know anybody that needs some local IA cleaned out, rocks moved, anything. I know I get 19 people I can call on to help me do that. But no, great job on the video, guys. You guys have been hard workers to every site we've been to this summer. So, you guys shine out there in the field. You can walk the walk and talk the talk when it comes to conservation, putting that sweat, blood, and tears into the aina. So mahalo, guys, for putting up with us and our sometimes false advertisements on the classroom. But yeah, I think that's all I have to say. Okay, well, we're gonna sign off, so aloha. Thank you for joining us, and stay tuned for next year's PIPES Symposium.
I know we're offline, but which is a good thing. I just want to say, we are? I was getting so emotional in the corner. I was watching you guys' video. I was like, oh my goodness, but it's just like my babies. They're growing. They're going to fly. They're going to leave the punana, And this experience has definitely made me remember what PIPES was like. This is exactly how I remember PIPES as an intern, It's just full of wonder, the ike that you guys are sharing with each other, that we got from our individual sites. I just remember PIPES being an intern. You guys put me back in that space, So mahalo, you guys. You guys are awesome. I love each and every one of you guys. Mahalo piha, mahalo piha, mahalo piha. Aloha.